So we will thank you again, all of you, for coming and sitting and asking questions, because that is what makes the session more interesting. Because you ask questions, then I need to think in a different way than what I've been thinking. And so now we'll continue. So yesterday we did innate immunity. Today we will do a little bit of antigen presentation, very minimal. I will not go into very many details. The reason I just want to give a little bit of a background about antigen presentation is because when we will talk about cancer immunotherapy, then this knowledge, you need to have a little bit of an understanding of antigen presentation. And then we will talk about T cell differentiation. Quite a bit of it is similar to what we had seen yesterday, but more from an innate immune system point of view. Today you will see how many different subsets and other things are there. So I think I'll start. So one thing I forgot to mention is most of the basic lectures which I'm giving are from QB Immunology. So I need to acknowledge that. But some maybe from Janway and others, but most of the slides are from QB. So okay, it looks better in my computer. But basically, why do you need antigen presentation? It's simply because when you look at the T cells, here we have shown CD4 T cells, but CD8 T cells also, they basically cannot see an antigen in its native form. If you have a protein, the T cells really cannot see it. So because they cannot see it, they will not be able to respond to it. So that is why you need an antigen presenting cell, which will process this, break down this antigen, and only a small part of it is presented, and that is what is recognized by the T cell receptor. So that's why you need antigen presentation. So these slides we will upload, and I have these links, so any of you, if you want to watch, these are links which will show you how this animation. See, because the T cell receptor, it can only bind. I can show you one slide and a little here. So the T cell receptor is organized in such a way that it can accommodate only a peptide maybe of 12 amino acids to 18 amino acids. So it's like this groove. So it cannot, there's no place for a protein to fit. The receptor, the binding site does not have enough space for a globular protein to fit. So it can only recognize small peptides. So that is why, if an antigen is coming in, most of the proteins are much bigger and they have like a 3D kind of a structure. So TCR does not have the ability to bind to those structures. So that's why, there's no place to bind. But B cell receptor has a place to bind. So, but T cell does not have. So there are two major pathways of antigen presentation. In what you have involvement of class 1 MHC, and in the other you have involvement of class 2 MHC. And in this case, you will get a CD4 T cell activation or helper T cell activation, and in this case, you will get a cytotoxic T cell activation. So the MHC 1 and 2 are basically, MHCs are molecules which have a place for the peptides to bind, and then they can show it to T cells. It's basically like, that's why it's known as a presentation, that they're presenting that peptide to T cells, but the processing inside the cell, the breakdown of the protein to peptide occurs inside the cell during the process of phagocytosis. In the lysosome, yesterday I showed you there are enzymes like lysozyme, low pH, so that kind of breaks down the protein into small peptides, and a part of it is then loaded onto MHC and then presented to the T cell. So what is the role of MHC in the immune response? The major histocompatibility complex is a cluster of genes which encode for this MHC1 and MHC2, and these are present on antigen presenting cells. So you see that in transplant rejection, it's the MHC which you need to match, because MHC, even though I'm just saying it's an MHC1 and MHC2, these genes are highly polymorphic. And so each individual has a different combination of it. Just to give you an example, so this is a mother and a father. They have different sets of MHCs, encoded by all these different colors. So now they have these offsprings. So you can have a combination of so many different ones, right? So if this one needs to have a transplant, it'll have a match here. That's also 50%. It'll have a match here, which is also 50%, and maybe 50% match from here. So even with a sibling, you don't have a 100% match. And sometimes you may not have this kind of a diversity. You may just end up with this and this, then there is no match. So this is a very, very, very basic kind of example, but because of so much polymorphism and different alleles of this gene, there are many, many MHC molecules, and that's why the MHC mismatch is so common. Even though there's so much of a diversity, generally it is seen that between races or ethnicities, certain MHCs are more conserved. So that's why there's more chances of finding a match within the same ethnicity than an outside ethnicity. Okay. So this is a slide which I already showed. So what happens is that the MHC basically has this peptide, which TCR has a binding site for it, and it can recognize. So one thing yesterday I had shown you that innate immune cells have a lot of receptors to recognize, antigens. So all innate immune cells have a similar kind of receptor. If it is a TLR, it will be a same TLR in all. The difference between innate and adaptive immune cells is the lymphocytes also have a receptor. All T cells will have a T cell receptor, and all B cells will have a B cell receptor. But each T cell, the TCR, is different. And that is why they can respond to so many different things. Because if they have a similar TCR, then if it is against a bacteria, then it should only be able to respond to bacteria. It should not be able to respond to a virus. So to take care of whatever it comes in contact with, the T cell and B cell have the receptor is different. So and this diversity is known as the T cell repertoire or B cell repertoire, like you have 10 to the power 8 or more than that. So that is why anything you come in contact with, there is going to be 1 or 2 T cells which are going to be able to recognize it. The other thing is that B cells, which we will talk in the next lecture, they can even rearrange their receptor to fit better to the antigen.
You're saying each and every cell has different, receptor
binding site.
Yeah. So which means it is keep on making mutations to create a specific receptor.
No, these mutations occur in the beginning in the developmental stage. So these two, the T cells and B cells have an enzyme known as RAG, or rearrangement activating. So basically, the TCR and BCR, when they are being made, only at that point these rearrangements occur, and each cell comes up with a different receptor. So it doesn't occur later. Only in B cell, as I said, there is more mutations, and then it optimizes better. But in T cell, no change.
How come the mutation is happening only to the particular gene? How it is?
Because I have not put this in the lecture, but if you will look, there is a VDJ region. So with that, the rearrangements occur. So there's many combinations which can occur. So that's a whole lecture in itself. I can cover it.
Why? How it is making mutation only in particular gene? It is not happening to the other genes. Because
the mutations are restricted to the binding site. Sometimes it is also known as in the antibodies the CDR3 region, because the genes are arranged in such a way that the mutations will only occur in that region. The rest of the portion is constant. Because signaling, see what happens. What the mutation you require is only where the binding site is occurring, because the rest of the reaction is same. You need to have signal transduction, same way, everything. Once the antigen binds, the rest of the process is the same. It's just the binding of the antigen, where the antigen should bind, which is different. And that is that is the beauty of the immune system, that it has evolved in such a way that you have these, it is able to generate mutations only in the binding region, and it is so specific.
Can we make this as a target for autoimmunity?
No. Because how will you make a target for autoimmunity? Tell me.
If we have a structure of this enzyme which is making mutation in those particular gene, so that it won't make antibody against it.
No. So even if you have that, because these genes will make random mutations, right? And after that, there is also a process of selection. When these different kind of TCRs are made, there's a process of selection, because it is made, say, 10 to 10 power of 10 to 10 to power 10 different kind of TCRs. Some may be reactive to your own proteins, right? So you have to make sure that those don't survive, and that is what my next slide is going to be. So if you will make for an autoimmune thing, again you need to have some kind of a selection, right? That so it's not as easy as it sounds, because you cannot direct what kind of a mutation is going to occur and what kind of a TCR is going to come, right?
Ma'am, yesterday you were telling now that stem cell therapy, they are using it for autoimmunity and all. So if we make, for example, type 1 diabetes. So for type 1 diabetes, it is degenerating only the beta cells. So if we make a stem cell, it express like PDL1 or CTLA4. So the immune cells will not touch that, and so the pancreas will be safe.
See, one thing you have to remember is, I don't think so. It's not only one mechanism. If the beta cell is destroyed, what is the point in putting a PDL1 or CTLA4? Because you have to start it as very early, and sometimes in type 1 diabetes, these are genetic defects which are visible, even in the infancy, when the immune system is not fully developed, because there's some problem and the beta cells maybe are not developing.
Same mechanism cancer cells are using, to escape from evasion.
Yeah, but there is a difference. In that, see, if you are going to remove the brakes of the immune system, like the PDL1 and CTLA4, if you're blocking, then you're going to get more of an immune response. So then you're going to get more of an immune response. If it is against a beta cell, you will get more of an immune response against a beta cell. So you will be having more destruction.
No, I'm telling we can put that brake so that immune cell will not touch that.
So then you have to so you want to say that you want to give PD1, right?
Yes.
But then you have to have a cell there which should be having a ligand for PD1. It should go and bind somewhere, right? Otherwise, how will it put a brake? So you should have some immune cell there which should be expressing either the ligand for PD1 or the ligand for CTLA4. Otherwise, if you're putting a cell which has a PD1 or CTLA4 on top of it, it has to go and bind to something to stop the immune reaction, right? So first you have to make sure that in that area you have to have either dendritic cells or monocytes which are so the other therapy is much more easier in that way, because stem cell you're giving one shot, it is regenerating your beta cells, right?
Yes.
So insulin will be made. So even if you stop the immune reaction, if the beta cells are already destroyed, it doesn't make a difference, because there's no cells to make insulin. So that is why the other approach is what is being tried more than this approach. We can say that, but in this case, it's not survival of the fittest. In T cell selection, like the TCR selection, it's not survival of the fittest. It's survival of non-self. Something which reacts to self has to be gotten, otherwise, if we are going to make T cells against our own proteins, we will basically be destroying ourselves, because there's going to be an immune response against our own things. And so in this case, it is not the survival of the fittest. It's more a survival of some T cells which are not going to be reactive to your own self. So this, I already told you that each of the lymphocytes expresses a unique antigen receptor, and somatic hypermutation has resulted in this diversity. So how do they get activated? So it's very simple. I had mentioned that the cells like dendritic cells are present, on the surfaces. So when they see an antigen, which is here, and then they start they become activated and they start migrating to the lymph nodes, which are the secondary lymphoid organs, which I had mentioned. Because the T cells are basically present in this lymphoid follicles. So once dendritic cells reach there, these are like T cells, which will bind to it. So, and then antigen presentation will occur and T cells will get activated. So, yeah. So this movie will not run, but this is using a technique known as two-photon microscopy. In this, what happens, you can do live cell imaging within a lymph node. So you can take out a whole lymph node. You already have cells, so you have mice which have dendritic cells labeled red because they're expressing a protein DS red or and in case of this, the green are T cells, which are already engineered that all the T cells in these mice have GFP in them. So now if you take out lymph nodes, you can put the whole lymph node under a microscope and you can see how the reaction is occurring. So this is something which is, there's a lab in our university, Dr. Cahalan's lab, Michael Cahalan's, and they do this very routinely. So in what they had shown is that you have a dendritic cell, it has an antigen on it. So now you have all these different kind of T cells, each will have a different TCR. What they do is they come, like they try to bind, they go away. They come, try to bind and go away. So he showed it very clearly, and if this movie runs, we can see it also. But if it finds something it binds, it stays there. The rest will, try to just come, go. So, and this whole thing is happening in a very fast manner. Like it's not like, okay, one is coming, going away, then the other is coming. It's not like that. It is like a lot of cells are coming, trying to bind. If they have something to bind, if their TCR fits the receptor, the antigen, they'll bind and the reaction will proceed. Otherwise, the rest will go away. And here you can see that some of them had been like bound and then the reaction is starting to proceed because wherever they're bound properly, you can see the yellow color. But this is just an example, to show that how people have tried to study all these things. So MHC molecules, so the antigens, if you think, they can come from outside, something which is coming from outside, extracellular. Also, they can be intracellular or endogenous because if you have a viral infection or a bacterial infection and the bacteria and virus are inside your cell, then the antigen is basically endogenous because it's inside the cell. Also, some of the endogenous antigens also be coming from your own cells if some things are being destroyed. But we will right now worry about only the foreign parts of it instead of the, the self parts of it. So there are two kinds of MHCs, MHC1 and MHC2. So MHC1 is basically supposed to present intracellular antigens, that is like viral proteins, self proteins, and even cancer-associated proteins. Okay. So, as I mentioned, this can help identify cells which have been which are infected with viruses and other things. And MHC2 molecules can present both intracellular and extracellular antigens, but basically they are supposed to present only extracellular antigens. And this combination only occurs in a specialized pathway known as antigen cross presentation, which is present in dendritic cells. So we will talk about that a little later. So class 1 MHC is present on all cells of the body. Nearly every cell which has a nucleus usually can express MHC class 1. The level of expression may be different, and sometimes during infection the level can go up or down also, right? So there is really no restriction. All cells will be able to present. And the reason for this is, if you see, what I'm saying is the MHC class 1 is supposed to present endogenous antigens. So it's not necessary that only macrophages have the capacity to take in viruses. Any cell can get infected with viruses or bacteria. It can be epithelial cells, it can be other cells. So all of these need to have the capability to present endogenous antigens. So that is why all cells have this ability. The MHC class 2, which presents the exogenous antigens, it is more restricted. And for these, it's only present on antigen presenting cells like macrophages, B cells, and dendritic cells. So the restriction for MHC2 expression is much more only to antigen presenting cells, because in this case, the kind of cells which get activated are CD4 helper T cells, which are not the kind of cells which will survey to see if some cell is infected or not. Their main function is to provide help to other cells. So that's why, and help to other cells to amplify the immune response in some ways. So in certain cases, some of these non-professional cells like fibroblasts, glial cells can also present antigens, thymic epithelial cells. These cells in certain cases can present antigens, otherwise, and this is MHC class 2 which I'm talking about, but mostly it is dendritic cells, macrophages and B cells. So this is just to show you that processing is required. So you have an antigen presenting cell here, and this is an antigen. So if you have fixed the cell means if you have added formaldehyde, formalin to it or paraformaldehyde to it, so it becomes fixed. Now none of the processes which happen inside the cell will take place. So it will no longer be able to process the antigen. It will take up the antigen. It may bind the antigen, but the antigen will not be broken down into small pieces to be presented to T cells. So if that is not occurring, you can see there's no T cell activation, right? In this case, you have no fixation, antigen is processed, and it's being presented, and you have a T cell activation. So you have a positive response. In this case, you have a fixed cell, but what you are doing is you are adding a processed antigen. So what you are doing is, instead of taking the whole antigen, you have already broken down in vitro in the lab, making some peptides, and that is done in some cases where you know, okay, which is the peptide which needs to be presented. An example is a lot of, you will see a lot of examples with OVA peptide, because that everything has been worked out. People know, okay, this 18 to 25, I'm just, this is random, it may not be the correct number. That is the part which is going to be presented by CD4 T cells. So they will make synthesized peptide, and you can add it to the APC. In that case, it will bind directly to MHC. It does not require any processing because it's already at the size which it needs to fit the TCR. But for this, you need to know the length of the peptide if it is going to be presented. So it is okay. It requires a lot of work because you need to map the whole repertoire for each thing. You have to do the whole binding. So it's not an easy thing that you can do for each and everything. But in that case also, you will get a T cell response because now the antigen is being presented to T cells. So here it just shows you that there is an endogenous pathway which results in antigen being presented by MHC class 1, and there is an exogenous pathway in which the antigen is being presented by MHC class 2, which I already kind of mentioned. So here it is. So then, I had mentioned that the MHC class, there is a pathway known as cross presentation. So what I mentioned is that the MHC class 1 can only present endogenous antigens. But in dendritic cells, there is an ability that they can take up the exogenous antigens from which are in circulating, and somehow there's this transfer that it is now presented on MHC class 1. And so it can activate, even the exogenous antigens can activate cytotoxic T lymphocytes. So this is an important property of dendritic cells which makes them a better antigen presenting cell, because so I can give you a little bit of details in the sense this occurs a little later than the other antigen presentation. Those are faster. It also requires, these are complexes which are used in case of MHC class 1, TAP complex, and this whole process basically is what is mentioned here is what is utilized by MHC class 1. And it needs to have antigen uptake because antigen is exogenous. If it's outside the cell, you need to have phagocytosis or something to take it in. So if you inhibit these functions, the antigen will not come in. So you cannot have a cross presentation. So if I go into too much detail, it is going to be very, very difficult. But the reason this cross presentation is important is it is very important for viral and anti-tumor responses. That's why so much of emphasis is there on DC based therapy, because it also has, it can do MHC1, MHC class 2, but it can also do this cross presentation. And amongst most, it's not that other cells can't do, other antigen presenting cells can't do, but the efficiency is much more in dendritic cells.
Peptide, the peptide is going to be fixed, that those amino acids or a peptide which will be recognized.
So there are many peptides generated, and many peptides can be recognized. The length is what is fixed. So I think it's either 9 to 11 amino acids for one MHC class. I'm not 100% sure. For other, it can go up to 18. So they can be, when you digest a peptide, there can be many which can be 18 amino acid, but not all of them are going to be able to generate some. So this is what is known as epitope. So some epitopes you can generate an immune response, some epitopes will not generate an immune response. So, and that is something you have to learn by trial and error. There are now predictive computer-based programs which can predict, okay, this part is going to be able to make, which may work, and then also you have to test, because predictions you have to always validate, right? Because they're based on some logical thinking, but not always everything follows. But it's not like there's going to be one peptide which is going to be recognized. So this is just showing you cross presentation, that the antigen is coming into this MHC class 1 also, and it's being presented. So this is just very small, like a brief introduction to antigen presentation. And now we will move on to how T cells develop. And this is also going to be brief, just to give you because if I go into details about all this, then I think we will have like 3 to 4 lectures on T cells only. Yeah. Okay.
Antigen presenting cells comes under includes the dendritic cells and B cells, macrophages. And this cross presentation of antigen is done by dendritic cells for both MSC and by MSC1. Do the B cells and macrophages also?
I think they can also cross present. Some people have shown, like it's not ruled out that they cannot, but it's not very much commonly seen in these cells.
B cells and macrophages. But it is more common in
dendritic cells. And it has been worked out mostly in dendritic cells. This presentation is relatively new, and most people have studied in dendritic cells.
But generally they do have a class 1 MHC because they are nucleated cells.
Yes. So dendritic cells in general, they can also activate CD8 T cells by their endogenous pathway. And they have MHC2, so they will also activate CD4. But in this case, what they are for the exogenous antigen, so they can do both separately or definitely. Yeah. But in this, they combine. They take the exogenous antigen and present it on MHC class 1.
Okay, when we come to dendritic cells,
yes,
simultaneously it can do both exogenous and endogenous.
Also. Okay. But in certain cases, not all the time does it do that. So as I mentioned yesterday also, that all immune cells develop in the bone marrow, and that includes T cells. So T cell precursor development occurs in the bone marrow, and then they start to travel and go to the thymus. And then here the selection of T cells, because I was mentioning that, so in the bone marrow you have a lot of T cells being made with different TCRs because of these mutations which are occurring, somatic mutations. But they can be against self, they can be against foreign, they can be against any different kind of things. So in the thymus you have a process of selection where so if you see when the antigen presenting cells, I was showing you that thymic epithelial cells also have the ability of present antigens. So in the thymus, you have dendritic cells and thymic this thymic epithelial cells. They present these self antigens. They basically self proteins or self peptides. And if basically the same process occurs as the antigen presentation, that the T cells which are being made, they will go through this, they'll pass through this presentation. If it binds, it will stop, right? So if it binds with if the T cell cannot bind, then it does not get a rescue signal. These T cells have specificities which the body doesn't think it is important, they'll die. So they need to bind to survive. So if there's no binding, the cell will die. If the cell binds very strongly, that means it has a very high reactivity towards self. Again, it will die because it's killed, apoptosis occurs. So their T binding has to be just right for those T cells to exit the thymus. Otherwise, they will not be able to exit the thymus. They will die. So they have to bind with a low affinity to low affinity to medium affinity. It should not be very high, and no binding will also result in cell death. And this is a body's way of checking that no self-reactive T cells are escaping the thymus.
So it is like a school for T cells. It is going there and learning what is self, what is non-self. So these thymus epithelial cells are expressing all our 18,000 gene proteins. Is it?
Most of the repertoire of self antigens are expressed in thymic epithelial cells and dendritic cells.
They're learning all our self proteins.
Basically, whatever cell proteins they can catch hold of and process. Okay. So most of the ones are there.
Which means the mistake should happen here only, no ma'am?
So that sometimes does occur.
Because if it is a recognizing cell and escaping from thymus, and it will go and attack our own cells.
So generally, the high affinity binding ones, very rarely has it been shown that they escape. It is more a problem of low affinity ones, which somehow can maybe even initiate, low affinity self peptide ones if there are more of them, or if there is somehow they are able to bind the self antigen. It can happen, but generally this process of selection is very, very.
But to understand autoimmunity, we should be studying this only, no? Then.
So this is so there are two ways, right? So one is, okay, T cells have escaped from here, self-reactive T cells have escaped from here, and that's why you're getting autoimmune. But that is one way, right? The other is sometimes many of the autoantigens are very similar to some viral antigen or something else. And in that case, some viral infection is generating a peptide which is similar to some of your self proteins. And you're now expanding your T cells for that, that virus specific peptide. And that also is now able to react to self. That also leads to autoimmunity. So there is never a very single, it's not like a straight line. One of the problems with immunology is it's not like, okay, this is what happens, will happen. No, it's not like that.
You're telling both good news and bad news, ma'am. I don't know what to follow.
This is why it's not so easy to develop immunotherapies or for so many of them to be successful. And there is a lot of people elute peptides, look at binding affinities, do all that. So a lot of people are working in this area also. So there are pockets of research going on. Some ultimately you need to understand a whole connection of it. What is really important? All of them are important. Also, it's not that okay, if this is happening, we have an answer. So this is one of the problems with most diseases. If it's so easy, we would find cures for all of them. So basically what happens is that final screening removes autoreactive T cells, and then the T cells are released into the bloodstream. And I think this is too much detail. Basically what this is showing you is there are two kinds of T cells. TCRs also, in which there are alpha beta kind of, the chains are alpha beta kind of proteins, or they can be gamma delta. Usually most of the T cells which you see in your bloodstream and others are all alpha beta. Gamma delta are very small and they are more innate immune like cells. So for the whole purpose of this lecture, think that they are alpha beta. So So far what we have learned is that the antigen needs to be presented by in the context of MHC molecules on antigen presenting cell, and then it will activate T cells. So it's not sufficient to activate T cells. That's the first signal, that you have the antigen presenting cell with the antigen, it's binding the TCR. So now the T cell and the antigen presenting cell have formed a synapse because they have this peptide in between, right? But that is not sufficient to activate the T cells. It's just kind of gives a signal, T cells become activated, but if they need to do further work, they need to divide and do other things, they need to have a second signal. And that second signal is known as co-stimulatory signals. So here is just an, like an overview in the sense that when the cells will become activated, the T cells will differentiate to do their functions. See, one of the things you have to remember is, so I'm telling you that there are 10,000, I just assume there are 10,000 T cells. They are all coming and searching for this peptide. You find one. One is not going to be sufficient to fight any infection. It's of no use. So now it needs to multiply that particular clone. So that is what is the next stage. That is what is the effector stage. The cells need to proliferate. So now you have like 100,000 cells of the same. So now they can fight the infection. It does multiply very fast. That's why, yeah. No, not half an hour. I don't think so. It takes one day or so to get enough number of cells. And so basically once the T cells become differentiated and activated, effector cells, then CD8 cells will become killer T cells. You have to remember this. They have the ability to kill, and CD4 T cells. Yesterday I showed you that there are different kind of subsets, Th1, Th2, etc. That is what the CD4 T cells will become, depending on what kind of a signal they will get. So here if you can see that the antigen recognition is occurring, which the antigen is being presented to CD4 T cell here and CD8 T cell here. This is an APC, you can call it dendritic cells or any cell, it doesn't matter. It can be monocyte and B cells also, but mostly it is dendritic cells or macrophages at this stage. And this, because it's getting the second signal also, the co-stimulatory signal, the cells are becoming activated. And at this point, they secrete a cytokine known as IL-2, which kind of enhances or amplifies proliferation, because they themselves have the receptor, if you see, for the IL-2 receptor, IL-2 receptor. So it's being produced, but it acts on these cells only. And these are all dividing, right? And you have these effector cells being made, and some cells will differentiate into memory also. And all this whole process takes place in the lymph node, because dendritic cells have the capability to migrate. Once they see an antigen here on the skin, the nearest lymph node, maybe this time it is near the shoulder, they will migrate. And yesterday I mentioned that they have a receptor known as CCR7. In the lymph node, you have the chemokine. So once they are activated, they express the receptor, it tells them to go there. So they go there, and that's where the T cells are. So this is the first signal, the TCR signal, which I mentioned, that this engages the T cell receptor to the APC. But it is not enough for it to proliferate, to divide. So then you need the second signal, and this is known as co-stimulatory signal, because this is kind of stimulatory, but you need co-stimulation for it to proceed. So this is why this is known as a co-stimulatory signal, and this is known as second signal. And the third signal comes in the form of the cytokines, which are secreted by the antigen presenting cells. And yesterday I showed you that cytokines which are being secreted by the antigen presenting cell, what kind of a cytokine, if they are secreting IL-12, that is what kind of decides what kind of a T cell response you will get, what kind of an effector cell will come. So all three signals are kind of required for a very efficient T cell response. So generally, if the APC is activated, you can, so yesterday I had mentioned like immature and mature dendritic cells, right? Immature ones express low levels of MHC class 2, even MHC class 1 for that matter, but MHC class 2 is what is more studied. It's lower levels. You have low levels of co-stimulatory molecules. But if you say activate them with something, you can see that the expression goes from, if you do by flow, it's here, it will go here. It's much more because now they're ready to present antigen. So these two molecules are required for presentation of T cell antigen to T cells. So co-stimulatory signals are required for T cell activation and proliferation, which is what I've been saying. So you have two kinds of co-stimulatory signals. The positive co-stimulatory signals, and these are present on T cells, CD28 and ICOS. And then you also have negative co-stimulatory signals, which will turn the activation off. And this is what we have been talking about, the brakes of the immune system. So because, and these are CTLA4, PD1, and this is BTLA, BT cell ligation attenuation receptor. But right now, these two are more in the news is what I will say. So why do you need negative co-stimulatory molecules? Because in all cases, when you start a reaction, immune reaction, you always need of these CTLA4 and PD1 is to shut off the immune response when it is not required. But the difference between CTLA4 and PD1 is CTLA4 comes very early in the immune response, PD1 comes in much later, which is something I will explain more in the cancer immunotherapy, and that will tell you why one therapy is better off than the other and why do we use the combination therapy also. By the B cells. So like usually it is seen that if you have the kind of T cell help which is provided, basically the cytokine response from the APC decides the kind of T helper cells which will come. The T helper cells will secrete a particular kind of cytokine that kind of decides the antibody response, that it will be more of an IgG2A or IgG, so IgG1, IgG2A, 2B or 3. That is, so everything is in a way dictated by the how the APCs are getting activated. So if there's more interferon gamma, then you will get Th1 kind of an immune response. You will also get IgG2B and 2A more of it, that kind of a isotype. And in the mucosa, you have IgA, right? And in the allergic, you have IgE. That is more IL-4 and IL-5 dependent. So Th2 cells will make B cells to make more IgE. So it is kind of decided by the T helper cells which are generated. So as I mentioned, CD28. So CD28 is present on T cells, but it needs to bind something on the APCs, right? So on the APCs, the two molecules which are the co-stimulatory molecules are the B7-1 or B7-2, which that's how they were known earlier, but now they have gotten their CD numbers, so CD80 and 86. And this is ICOS. So usually ICOS is also a co-stimulatory signal and a positive one, but it is more usually in the later stages of the immune response, more in the memory and other things. Usually initiation on the beginning, it's mostly the B7-1 and CD80, 86 and CD28 interaction, which is important. And so same here, we are just showing you the inhibitory receptors. This is program cell death receptor, so cell death 1. So, and this CTLA4, I don't even remember the co-stimulatory something, I don't remember the, so now see, if you look here, CTLA4, it also binds the same receptors, CD80 and 86, which the positive co-stimulatory molecules bind. Same. The only difference is in the beginning of the activation, you have this being present on the T cells. By the third day, when T cell has already, CTLA4 starts to come up. Once the activation is done, CTLA4 comes up because now it is going to be used to shut off the immune response. PD1 does not come up that fast. It comes up much later. In fact, it has been shown that when the cells, there is a chronic activation because you have a low level viral infections, or you have some other kind of long lasting chronic infection, then the PD-1 comes up. So it's a much later kind of a break which comes on the T cells. That's why it does not cause as many problems as CTLA4, because this comes up very fast in the immune system, and it plays a more major role in breaking. So this is what I already mentioned. Okay. So as I mentioned, that it is induced 24 hours after activation and peaks by 2-3. And the reason it is able to, oh, I forgot to mention this, that this co-stimulation is already going on, like B71, B72, CD8086 with CD28. So why will it now suddenly bind CTLA4? CTLA4 has a higher affinity for it, so it displaces CD28 and it goes to the T cell now binds via the CTLA4. So this is just kind of a summing up. If there's only signal 1, here you get anergic. Basically, anergic T cells are T cells which sometimes remain in the body for some time and then slowly they will die, but they cannot make an immune response. They may not die immediately, but they will die. Anergic means they are on their way to cell death. Okay. And in this case also, there is a negative co-stimulation, the cell will die. So only when you have this MHC and then you have the positive co-stimulation with CD28 and CD8086 along with the cytokines, you have an activated T cell and an effective immune response. The T cell will die. Yeah.
Immune response. If it is sort of, then T cell will die.
Yes, the T cell will die, because the T cell, if it is shut off, that means that the whatever the T cell was supposed to do, it should have done, and it's no longer required. See, because at the beginning of the immune response, what you have done is you have taken that one T cell and multiplied it into 100,000. So that means there is now not enough place for other T cells. So, and once it has taken care of the infection, it needs to get rid of all these cells and come back to that one. But maybe because there's memory, you don't have one, but you come back with 10 to 12, and those are stored as memory cells. So that's why the second time around, you are much better able to fight infections, because instead of one cell, now you already have 10 cells there, which can recognize that particular infection. So this is, I've already mentioned that IL-2 is kind of important. Some people even measure proliferation by measuring IL-2 levels. If the IL-2 levels are high, then they think that cells are proliferating. If you have no other means of looking at proliferation, there are many, many ways you can look at T cell proliferation. One is simply, if you see the cells will divide, DNA will, you need to have DNA synthesis and division. So you can take, one of the bases is thymidine. It is taken as a radioactive base, and it should be incorporated more and more. That is one way of measuring proliferation. You can measure by flow cytometry also, which I'll show you in the next lecture. So many, many ways people have devised. So this is kind of a summing up, basically. So I will kind of leave this, because I seem to be saying this over and over again. So hopefully by now it has been clear. And so we will move on to this. And this I had already talked about yesterday and shown you examples, that the cytokines which are secreted by APCs will dictate the T helper cell differentiation. What kind of T cell will be made. And this is what I showed you yesterday. An example is simply that viruses, if they induce the production of IL-12 in the APCs, you get Th1 responses. Th1 are T cells which will produce interferon gamma. If you have a warm infection, it gives IL-4 and you get a Th2 kind of an immune response. I showed you examples of it yesterday also. So this is basically showing this in a more pictorial form, you can say, that you have in the T cells, basically you have also transcription factors which decide it will go into Th1, and these cytokines activate these transcription factors. And once the cell is, okay, maybe here, so that's, I thought it will be easier. So if the APCs are secreting these kind of cytokines, you get a Th1 response. So these T cells will express a transcription factor T-bet, and they will produce these cytokines. And these are the major functions, that they are more involved in protection against intracellular pathogens. And the interferon gamma can activate cytotoxic T cells. And the interferon gamma can go back and enhance macrophage activation, so enhance APC activity also. Similarly, in case of Th2, this is the cytokine and this is the transcription factor. And these are the cytokines which are secreted by Th2. And if you will read, these are the cytokines which are involved in allergies. These are cytokines which become high. And these also induce secretion of IgE type of antibody from B cells. So this Th subsets, the numbers seems to be increasing day by day. We started with these two. For a long time, there was only Th1, Th2. Then we included, this Th17 and Treg. These two came, and more recent are this Th9 and Th22. And T follicular helper cells are more involved in where the B cells are making providing help to B cells to make antibody. So they are present only in a specific reaction known as germinal cell reaction. But most of the others are involved in protection against some kind of the organisms. Like Th17, I had mentioned yesterday, they are better. They provide protection against fungal infections. The Klebsiella pneumonia infection is also requires this Th17 kind of an immune response. If you don't have this immune response, you are more susceptible to the Klebsiella pneumonia infection. And these cells are highly inflammatory, so they are also involved in autoimmune diseases, in fact, like multiple sclerosis and others. And these new subsets, the Th22, these have come up more when in the mucosal immune system, like in the lungs or in the gut. So those studies are more recent, so that's why we have now come up with this new subset Th22. Basically, the number which is there, apart from 1 and 2, it kind of tells you the cytokine it makes, the later ones. Th17 will make IL-17, Th22 will make IL-22. IL-22 is important in protection in lungs and all, because the receptor is expressed on epithelial cells, and it helps the epithelial cells to multiply, produce antidefensive peptides. So that's why it is more important in this mucosal immunity. So this depends. A lot of labs focus on only one kind of subset and study that. So this is just showing you that how each one has a different specific transcription factor, and that is some way, in one way you can use it to recognize, apart from, because all of them will express CD4, right? Only the cytokine which is being secreted is different. So how do you know from that, okay, this is a Th1 cell? You can look at the cytokine profile and say it is a Th1 cell. You can also do a staining for the transcription factor and see if the transcription factor is also showing you it as a Th1, or you can do a PCR, polymerase chain reaction, to see if this particular transcription factor is upregulated. So this is more used for that kind of a thing. So this, I think I have already mentioned, so we will move forward. So this is, see, if you have a Th1 kind of immune response, it will inhibit the Th2 kind of antibodies which are being made. You were asking that what kind of, so if you have a Th1 kind of a T cell which is going to make interferon gamma, you will not get an IgG1 antibody or an IgE antibody. You will get an IgG2 antibody. So similarly, this one kind of, there's an active inhibition of the other side. So it's reciprocal. If you will get Th1, you will not get Th2. Similarly, in case of Th17, it's more reciprocal with T regulatory cells. So if you have high Th17, you will have low Tregs, the other way. But this commitment to these lineages is not absolute. You still have plasticity. You can go back, but less easily once you are committed. But it's not still absolute. So that's why these transcription factors are known as master regulators. So once they start being expressed at high levels, the cell is more and more committed towards the kind of T helper response it is going to make, because it actively suppresses the other gene. So if Tbet is expressed, it will start suppressing GATF3. That is the Th2 transcription factor. And this is what I already mentioned. So what is funny is that TGF beta, a transforming growth factor beta, is a cytokine which is involved in both these subsets. So this one is highly inflammatory. This is, as I mentioned, it showed you yesterday also, this is a suppressive T cell. Both of them require this TGF beta, but IL-6 basically decides. If you have TGF beta along with IL-6, you will get more Th17. If the IL-6 is absent, you will get more Tregs. So it's all these cytokines. That's why they play an important role in deciding the fate of T cells. So this is just to show that the balance between two is always ideal, because you have optimum amount of inflammatory response and optimum shut-off responses. Whenever this balance is disturbed, it leads to some problem or the other. So at homeostasis, basically you need to have a balance, but in case of infection, you need to, if it's a bacterial like pneumonia infection, which I mentioned, you need to have more of the Th17 and less of the Tregs. Otherwise, you will not be able to fight infections. So this subset I had mentioned, so this is generally not involved in fighting any kind of infection. Its major function is to enhance B cell function. It enhances the differentiation of B cells for antibody production. That's why this subset is important in autoimmune diseases where you have autoantibodies being made. This enhances the production of those B cells. But otherwise, it's not important for any kind of this fighting infections. It's important in the sense it will activate B cells, but by itself, it does not fight any infections. So this is a kind of an overview where you have an antigen presenting cell, and it is activating both T helper cells and CD8 T cells. These are proliferating, and you have perforin granzyme, you have memory generation. B cells can also present this antigen now and making this plasma cell. So this T cell which has become activated now provides helps to B cells. And now these B cells will divide and make antibody. This is like a very broad overview of what happens in the, and this I already mentioned that the commitment is not absolute. It can revert back. And that's why, if you're wanting to say that this is a Th1, you have to do multiple things. You have to look at the cytokine, you have to look at Tbet levels, you have to look at GATA3, because if you're saying Tbet is high, then you have to show that the other one is down. The other four are all down. Because sometimes now you have cells which are like Th1, Th17. So you will have expression of two transcription factors. Especially in tumors, we are seeing more of these kind of things. So this is just an example that you have two kinds of leprosies. One is tuberculoid and one is lepromatous, right? In the response in both are different. In one, you have more of a Th1 response, and the other you have more of a Th2 response. And this can be shown in the next slide. So, okay, let it ring. So the Th2 cytokines are upregulated in this leprosy patients, and you can see here the Th1 cytokines are up. So even in the same disease, you have different presentation depending on the phenotype of the disease. So now we come to T cell memory. So you have three different subsets. They are known as TCM, T central memory. CM is central memory. TEM is effector memory. And then this TRM is a newer class of T cell memory, which is known as tissue resident memory. These two have been studied for decades. This is more recent. So basically, the TCM comes before TEM, and people believe that one kind of memory is more effective than the other, and different kind of diseases will give more of TCM or TEM. Again, that's a very, very wide field, and this is also very important to study because T effector memory differentiates very fast into effector cells to fight infections on second exposure, while TCM will take comparatively much longer. So people are interested in maybe generating more of this kind of memory for more problematic infections, so that they can, which proceed very fast, so that you need more of a T effector memory so that it can immediately get activated and control the infection. So in fact, still being worked out how you can get one kind of a memory more than the other. Some things are becoming known. That's why so many different adjuvants, are also being tried. That you try. Yesterday I mentioned that you use parts of LPS. Then they want to see that, okay, it activates APCs, you get activation of T cells, but what kind of a memory response is it generating in the end? Is it good for us? So a lot of combination of these things are being tried as adjuvants for vaccines. Basically, wherever you want to generate memory, is this memory better? The T effector memory better for this or TCM? So still people are trying to work out signals, how we can get one kind of a memory than the other. And sometimes we have seen that in certain cases, like I think it was smallpox, that you see that the memory lasts for 25 years. You don't need to give a booster. In many times you will see that when you are vaccinated, in 10 years or in 5 years, you have to get a booster because the memory is such that it fades over time. So you need to get boosters so that you get again a generation of memory cells. But smallpox vaccination was given once, but you are protected for life. So the memory which is generated in that case is so robust that you don't require any booster. Huh? Tetanus, it's in 10 years you need to have a booster. Every 10 years you need to have a booster for tetanus. Yeah. Because they have kind of realized by looking at the titers of antibody in the serum that now you no longer are able to see that titer. So if you give a booster, so that's how it has been worked out that now you need a booster. So by 10 years in tetanus, it is 5 to 10 years. BCG is more long. Yeah, and the other case, BCG is only protective against in infants. It doesn't work in adults as much. So that's why you see that booster is not given only because it's given just after birth, BCG. Only in developing countries. It's not even given in developed countries because they believe that by that you are giving the bacteria, and which may sometime later on cause dissemination of TB. So as I mentioned, T effector memory is the most effective because the name as it suggests it results in effector cells. Then tissue resident memory has come more recently, and what people have realized is that, okay, we talk about these memory cells, but they need to also be present in tissues where the infection will come. So like if influenza infection will come via the nose, right? If you inhale. So these tissue resident memory cells are present in the lung. So that's why now when they've started doing this mucosal immunity, they realize that these cells which are in these small area niche where the infection comes, they are more important than the ones which are probably residing in bone marrow or spleen or lymph node. So this tissue resident memory, that's why has suddenly gained a lot of importance. And most of these are CD8 T cells. So this is just to show you that you have first a T central memory cell being made. A part of it is stored in as memory here, and then effector memory comes. Part of it is again stored here, which can differentiate into two effector cells, and then differentiation of T cells, multiplication of T cells, and ultimately death of T cells. And these both cells can result in this T resident, tissue resident memory cells. They can also come from T central memory cells. This is a relatively new area, so still people are working out how this tissue resident memory is generated. As I told you, there is a whole field of immunology which is focused on how you can get the best memory or the kind of memory you want. So is there any difference between CD4 and CD8 memory T cells? So generally you have more of CD8 T cell memory cells than CD4. And they also proliferate more during their responses. So it has been worked out that the memory CD8 lifespan of memory CD8 T cells is longer than CD4 memory. I think I read somewhere that in general it is like, they've looked at a few ones, if it is 54 days for CD4 T cell memory, it is 132, like it's nearly double for CD8 T cells. So that's why they are more longer living. So the other thing is, as in many cases, I told you that the memory cells can live for very long, even 20 years, people have shown. So there's no antigen there sometimes, at least not what we can see. So how they survive? So that is still something, a gray area. It is definitely known that IL-7, one of the cytokines which are secreted by stromal cells, helps in maintenance of memory. But so what is believed is some signals are given that in between these cells proliferate and again become memory. But not always an infection is required for this. So as I said, that the whole of this T cell memory field, quite a bit is known, but still answers are not well defined. So far I have shown you that you need to have antigen to be processed, then only it will be presented. There are certain antigens which are known as super antigens. What they do is they don't need to be processed. They can bind directly to the MHC, if you see, alpha chain of class 2 MHC. And they can be exogenous or endogenous. And because they don't require processing, they are known as super antigens. And examples are, you will see a lot of these toxins. This is Staphylococcus toxins, which cause food poisoning. These are all, they don't require any presentation. That's why they are so highly, the response is so fast, because basically they will go and bind the APC MHC and they are present, they will activate the T cells and lead to all these problems which we see. Most of them, if you will see, are toxins. But it's not, in this case, there are also some other things, but mostly it's toxins which act as super antigens.
Why do we vomit when we get food poisoning?
I think vomiting is more to try to get rid of that particular toxin. I don't think it is related to an immune response. It may be playing a part, but it is more also a part of the digestive response that you vomit. Yeah, definitely.
There is a CTZ center in our brain, the chemotrophic zone, something like that. I can't recall the full form. That CTZ center actually detects the toxins. So in our brain there is a blood-brain barrier, right? So it is seen that over that CTZ center, we essentially do not have that blood-brain barrier. So the toxins
Can go in.
can go there, and that then it stimulates the response that now you have to vomit. The contractions, it stimulates those
So it
contractions.
kind of binds there, and then
Yeah.
it'll give the signal that now you should vomit.
Vomit.
Maybe it's a protective mechanisms for these. It has
Yes,
evolved
yes,
in
ma'am.
such a way that you don't need the blood-brain barrier for this to cross and bind to that
Yes, ma'am, because
center. Yeah, to some are reflex actions in a way, you can say. Yeah. Thank you. So some of these slides which I show you like this are overviews, in the sense like if you want to see the whole thing, you can just look at this slide and it will tell you what I've talked about this far. So in this, it is simply showing that you can have a sequential activation, like one APC is activating helper CD4 T cell, and another APC is now activating this CD8 T cells, and then you have CD8 T cell getting activated, and you have a memory cell. In some cases, you can have one APC being activating both cells, and ultimate result is the same. So it is not like there is no, in many of these things there is no hard and fast rule. It's not nothing. It's not absolute. Like, okay, first it has to bind CD4, then it will bind CD8. It all can happen simultaneously. It can happen sequentially. Basically, it can proceed however it suits the antigen. So I had already mentioned this cross presentation because it can activate CD killer T cells. See, killer T cells, why they are considered very, very important is they will kill any T cell which is infected, any T cell which is transformed by tumor cells. And these two diseases seem to be more difficult to fight. That's why there's a lot more emphasis on understanding how CD8 T cells can be generated and how you can, because they have the capacity to be able to lyse the other cell. You don't have any so far. There's no really good therapeutic against influenza virus, right? What you're supposed to do is take rest, eat, drink fluids, and it'll pass away on its own. But you don't have any kind of medicine which you can take because there are really no good antiviral compounds which are known. So this I would not worry about TC1, TC2, because what has happened in the field of immunology is every time any cell is there, people divide it into hundreds of subsets based on the cytokine, surface expression, but ultimately all of them are, if it is a cytotoxic T cell, it is going to kill. Maybe it will kill this one better than the other one, this subset is better. But that is like if you're really going to do a lot of research in the field, you worry about that. Otherwise, just cytotoxic T cell is the one which will kill is enough to understand. So this is just to that you can look at antigen specific T cells using this MHC tetramer technology by flow cytometry. So people do this for cancer cells and also for virus. They have made these tetramers, which are like peptides which will bind to the MHC, but they have put a fluorescent signal on it. So you can see in the whole CD8 cells in your body, if you have 100,000, how many of those are specific to your virus or specific to your cancer. So that's one way of tracking, because you always want to know that are you generating an immune response against, suppose you are giving a vaccine against a virus. Now you want to know if it is resulting in, you want T cell activation and B cell antibody secretion. B cell antibody secretion you can look in the serum, look at the antibody titer by different methods. But T cell activation is more difficult to follow, especially antigen specific, because you can look at markers which get upregulated on T cells on activation, but it doesn't tell you that it is antigen specific. Maybe something else is there which is activating them. So that's why people, when you want to follow antigen-specific T cells, this is the technology which people use. Especially, it works for CD8, and now there are tetramers which can be used for CD4 also. But it is not as easy, because making these tetramers and finding the right tetramers is a whole job in itself. I think we should. So this is generally what happens if you have an infection. The T cells will expand, and once the virus is gone, you have removed the infection, then the contraction should come. You need to get rid of. So at this point, at the contraction phase, there is massive cell death of the T cells which have expanded. A lot of apoptosis undergoes, and few cells remain as memory. So you have started from here, like you had one cell, and now you have more than more than, this is days post-infection, but as you can see, you started kind of from zero, but now you have many more T cells against the virus, and that is what is memory, basically. This I think I did mention yesterday, but how the T cells, cytotoxic T cells kill the other cells, there are two major ways. One is directional release of granule contents, that is via fast ligand interaction. And so as you can see, there is a kind of a conjugate formation between the target cell and CTL. And because of this, there is this release of these granules, and the cell, the target cell, which is orange here, dies via apoptosis. So the granules are kind of released only in that cell. So it's a directed release, and it is not secreted out in the milieu or anything like that. So this is fast ligand interaction, and then the other one is what I mentioned yesterday, the granzyme perforin mediated cytolysis. So in this, first it makes this perforin which creates the pore, and the granzyme, which are serine proteases, are pushed inside the other cell. So basically in both cases, you are putting granules which have proteases and other things which are going to digest the content of the other cell type and kill it. And in this case, because you are forming the pore also, the contents of the cell will leak out, so the cell will die. So this basically sums up what I've shown, and I already mentioned that it is a much more gray area in many ways. So people have different theories, and they stick to that theory. There's a lot of there's no clear-cut direction for this at this stage, that we can say, okay, this is how this is made. No, each lab, how it functions, what kind of a system it is using, they have their own defined pathway. So that's enough for T cell biology for now, and maybe we can, since we have time, I don't know, is it already 12 o'clock? Oh, okay, 12 o'clock. We can start a little bit of B cell and maybe then go to lunch, or maybe we can go for lunch and then we come back and do B cell, whichever way you all think, it's fine. I have, in B cell lecture, I have much less number of slides. It should not take more than half an hour. That's, Because what I was thinking is that after lunch, today we will just talk about how the different methods, how you study immunology, what is like how you use flow cytometry to study immunology, how you use ELISA and measurement of cytokines and other things for.
Cell replication of mycobacterium is one of the problem for the treatment, but secondarily the macrophages, which is not identified by our immune system, is that the problem of defending against the tuberculosis?
So the tuberculosis bacteria basically remains inside the macrophages only. Like it uses the macrophage as a reservoir, and the problem is sometimes that macrophage does not show any signs on the surface that it is having a mycobacteria inside. So it is, even when NK cells, natural killer cells, are surveying, or even cytotoxic T cells for that matter, they don't see any difference. So they don't kill that particular cell. So that's one way how the tuberculosis bacteria survives. But your question was that.
No, interconnected question is that after the taking of the therapy for a more than longer period of time, after the latency and afterward 4 months, later on it will be we can think that the body get acquainted with the number of antigen representing complexes. So in future there may be a chances once again to get infected with the mycobacterium, and till then also the body is not recognizing particular this type of the mycobacterium. Is it the failurity of the macrophages or the transcription which is taking place in a macrophages?
So when you have the therapy, it's supposed to get rid of all the mycobacteria. That's why the therapy is so long, right? So there are two things which can happen. Either some mycobacteria are remaining and that's why they come back, or you get another infection of TB and now so this therapy doesn't make you resistant to mycobacterial infection, right? It only kills the bacteria which are there at that time. So it's not like a preventative therapy. Like it's not a vaccine that once you've had it, that you won't get the infection again. So that's why I think it is possible that even if you've had the therapy, you are still can get the infection. See, memory is not generated. And in many of these cases, in malaria and tuberculosis, definitely one thing which has been seen is that generally you make memory against, immune response is generated against the surface proteins which are there, right? And in malaria also and in tuberculosis also, the surface proteins which are most abundant on the surface, or you get a very good immune response against them. Generally the body makes, but that response doesn't seem to be protective. So what is also now being believed is that overexpression of these, certain kinds of surface proteins is kind of a decoy which is used by these kind of organism, that all the immune response is directed against these, because so many of these proteins are present on the surface, that but this response is not enough to kill the bacteria. You have a very high immune response, everything, but the bacteria still survives happily. So this immune response is not effective against this. So now they think that there are cryptic antigens in these organisms. Tumor cells also, people think this. Earlier people would look for one tumor specific antigen and try to make an immune response, and many a time they were successful in making an immune response. But the tumor, there was no effect on the tumor. Tumor was still growing happily. So it's not necessary that if you have an immune response against an organism, it will get rid of the organism. It should be against the right part of the organism. Maybe it doesn't interfere with the replication or anything, this response against this protein. So that's why so many initial attempts were all focused on like circumsporozoid protein of malaria, but none of the vaccines worked. So now we are moving into B cell activation, the other lymphocyte of the adaptive immunity, and this is just a germinal center reaction, where B cells are able to multiply. We will come to that a little bit. So immature B cells. So immature is a term which is means unactivated B cells. Okay. So B cells bear receptors which are basically antibodies, which you see in your circulation, are the B cell receptors. Only in this case they have a transmembrane domain, so they are anchored on the, they are displayed on the surface, and then once this transmembrane domain is gone, it secretes. So when antigen stimulation is there, the B cell will become activated. They will also go to the lymphoid organs, or they are already in the lymphoid organs. They're be in lymph node where they will multiply and secrete antibodies. And once the reaction is over, you have more of these B cells of the same receptor remaining as memory cells. Okay, and same as T cells, B cells also which react to self are deleted during development. Only difference is the T cell for T cell development, this selection occurs in the thymus, but for the B cells, the selection occurs in the bone marrow itself, stromal. So it's basically same as T cells. I told you that each of the receptor which is present on T cell or a B cell is different. So you have all these, 1, 2, 3. Here you can see all these are B cells with different kind of specificities of B cell receptor. Here it is reacting to self, so it is deleted. So an antigen comes, it's binding to the second B cell, so that second B cell is expanding, and now you have many more of those B cells. So basically in both cases, what you are trying to do is, in case of T cells or B cells, you are trying to find that particular antigen specific cell, and now you make that clone expand, because one cell is not going to be enough to fight any kind of infection. So you make it expand, so now you have enough numbers to fight, and once everything is taken care of, the infection is gone, you now get rid of all these maximum, huge amount of cells that you have made, because you don't need them. So they die by apoptosis, but not all die, some will remain as memory cells. So you started with 1 cell, now you have 10 cells. So that's why next time when the same infection comes, you already have some cells which can now respond. You have more than 1 cell now which can respond. So response is faster. And the memory cells require less signals also. That's why they can proliferate faster. So B cell responses can be divided into 2 different kinds, T dependent and T independent. So I had already shown you in the last lecture that one of the major functions of CD4 T cells is to provide help to other cells, help to B cells, help to CD8 cells. So all the T dependent responses will require that help from T cell, but certain antigens can activate B cells directly and they do not require T cell help. That's why they will be known as T independent. So process in both cells is more or less the same. Once the receptor is engaged, then signal transduction will occur and these cells will proliferate. And same thing occurs in B cells also. The only thing you can say is B cells, this proliferation occurs in special areas in the lymph node known as germinal centers. And in the germinal centers, there is a T helper subset I showed you, T follicular helper cell is also present. So it helps B cell proliferate, it helps B cell differentiation. So this germinal center is basically an organization of cells. It's nothing more than that. So basically what the T helper cell does is it provides these signals, co-stimulatory signals. They're not co-stimulatory signals, but cognate signals in terms of like CD40 ligand interaction. So this, and they also secrete cytokines which help B cell proliferate better. And basically B cell proliferation, activation, the T cells provide help. Okay, can you see this? Okay, so you have a mice, and if you irradiate them, you basically are getting rid of all the T cells and the B cells, right? And then if you reconstitute the mice with bone marrow cells alone, you don't get an immune response. If you reconstitute the mice with thymus cells alone, so if you reconstitute with bone marrow alone, you are getting only the B cells. If you're putting only the thymus, you're putting only the T cells, there's no response to the antigen if it's a T dependent antigen. So only when you have both that you have an immune response. And this is how people worked out, that you require T cell help for B cell responses. So the initial receptor, which is there on the B cell, is known as an IgM receptor. So this is also an isotype of it's also a kind of an antibody. The thing is, it's supposed to have an affinity for the antigen, but the affinity is not supposed to be very high. But instead, this IgM is, once it is secreted, it's a pentameric structure. So it can bind more antigens. It has more avidity, but not very high affinity. But the B cells in the germinal center, this whole thing is arranged because in this, the B cells can again undergo this mutation, a process known as somatic hypermutation. So they kind of rearrange their receptor. So if it is binding, say, this is a binding site and it's binding, like this. Now it will mutate this receptor so the binding becomes like this. It becomes tighter, more higher affinity. So because what you want is it should be able to bind with high affinity to remove the antigen more efficiently. So that is why the B cell memory which remains is not the same as what the cell you started with. The cell which remains as memory has a higher affinity for the antigen because of this somatic hypermutation which takes place in the germinal centers. And in the centers, the antigen is also presented by follicular dendritic cells. But I think the amount I have told you, just remember that the affinity of the B cell receptor for antigen increases over time, and that is the most important thing you should remember. So in all cases, in all cells, if something binds, the cell becomes larger. And that is one way of looking at activation. It doesn't matter. Here I'm showing you for B cells, but the size will change. It becomes bigger. And then basically an antigen, once it comes, it binds to many B cell receptors on the, there are many B cell receptors, all with the same specificity. So there's cross-linking and then the signal transduction occurs. And this is what is simply shown here. It's not anything better. And this is basically showing you kind of a timeline. Okay, so antibody secretion is visible by 4th day to 5th day, about in a week's time. That's why the adaptive immune response comes.
T cells are getting trained by thymus epithelial cells for
getting selected by thymic epithelium.
Yes. What about B cells where it will get
bone marrow?
Bone marrow.
Bone marrow itself, it gets selection. So you see, in the naive B cells, when the B cells have not encountered any antigen, they have their surface IgM, right? And when they see an antigen, they secrete this antibody. So if you want to look at very early immune response, you will see IgM antibody in the circulation. The isotype of the antibody is IgM. And then once the germinal center and other reactions have taken place, you will see that now you start seeing IgG antibodies. That is what the antibody you see after 7 to 10 days. That's why it takes about 7 to 10 days before you can start finding antibodies in the serum. And this, the process I already mentioned, which happens in the germinal center, and this is an enzyme AID which is involved in this somatic hypermutation. And there are reports which show that some of these, there are some patients who have deficiency of AID or a mutation in AID. So then the B cells cannot switch, because now you're switching from IgM to IgG, and also you are making a higher affinity B cell. So that process will not occur if this enzyme is absent, the AID enzyme. So this is more for people who are working in the field, that the molecular weight of these IgGs of different isotypes are different, and serum levels are also different. You will see that the serum levels for IgA is generally high, and IgG2 is, these IgGs are high, and and IgA is generally high. Its baseline of IgA in the serum is high. Its half-life is, for IgGs, is about 3 weeks, so 21 days to, and that is why, yesterday I mentioned that in some people who cannot make antibodies, you give this infusion of intravenous immunoglobulin. You have to do it every 3 to 4 weeks, because by then it is removed from the body. And all these can activate complement, because antibodies function in many, many different ways. And that is kind of summarized here, that they can bind directly to antigen and remove them. They can bind to viruses and neutralize pathogens and toxins. They can, okay, maybe I will just use. They can also enhance phagocytosis, because they can, the complement receptors, when they sit on the surface of these bacteria, which was shown yesterday, they leave a fragment. It kind of marks these bacteria to be removed. The antibodies can bind to that also. They use, it enhances the binding of antibody for opsonization. And the antibody molecule is a Y-shaped molecule. The lower portion has a binding region for this Fc receptor. So that is how the antibody can bind to many other cells and do different functions in them. The Y-shaped molecule is like this. These are the binding sites, the top part, and this is the constant region, which is binds to the Fc and do a number of functions. And this, what I mentioned, that most newly generated B cells, the ones you have expanded, will disappear once you have gotten rid of the infection, and a few will remain as memory. And so, because of the germinal center reaction, now you have higher affinity antibodies being produced, which are better able to neutralize the antigen. So this is, as I said, this lecture is like a very short lecture. I think we will finish in 5 minutes. Okay. And so many of the plasma cells, so B cells, when they start secreting antibodies, they no longer have this on the surface, because now it's that same molecule is being secreted out. And at the stage, it is known as a plasma cell. And this plasma cell, some of them have been shown to be very, very long lived. Even like 10 to 20 years after that people have found it in bone marrow, that these are, cells which are generated, that these are known as long-lived plasma cells, which can produce this antibody over a period of time. So I think I've already mentioned this, that the memory cells have already undergone affinity maturation. See, affinity maturation is the process where you rearrange your receptors and you get higher affinity receptors. So your memory cells will have a higher affinity for the antigen. This also, I think I have shown it in a different slide, but if you want to see, then the antibody will appear in the serum not before 7 days. In most cases, there's always exceptions. So 7 to 10 days is when the response is supposed to peak. And that is why you need to have the innate immune response to take care of the infection in the first part, so that by the time this response develops, the infection should not be so high that, okay, this is of no use. So so here, what I mentioned, the affinity of the antibody increases over a period of time. So some people say that there are also IgM bearing memory B cells, and this has been shown in some diseases, that some of the memory cells which develop are also from the, in the very beginning, when they have not even switched to IgG, or they have not undergone affinity maturation. So you have both kinds of memory, IgM bearing memory cells and IgG bearing memory cells. Generally, the IgM bearing memory cells are lower in number. It's mostly the IgG bearing memory cells which are supposed to play a major role. The IgM bearing memory cells are shown in some, have been studied in more in humans in some patients and other places, in some diseases where they seem to play a role, which I will show you in the flow cytometry lecture. So T-independent antigens, basically, you have nude mice. In these, there is no thymus, so there are no T cells, basically. So, but still, if you give an antigen, you can see an antibody response. So that means there are antigens which can generate an immune response in the absence of T cells. So those are supposed to be T-independent antigens, because now in this case, you're not needing a T cell.
It takes 7 days to get a good amount of antibody to contain
at least visible, yeah.
Yes, but like a disease like Ebola, Nipah virus and all, by the time 1 week, if you leave it like that, patient will die. So, and that condition, we should be giving monoclonal antibody against that virus or bacteria, so that we can at least minimize the death rate.
See, 7 days is when you start seeing it in the serum, in the sense, for your methods of detection, that at that point you are starting to see. But the antibody response probably comes a little earlier. And also before that, you also have the innate immune system, which is already trying to contain the virus and bacteria. So monoclonal antibody, see, I don't have it, I don't think I'm going to talk about in this lecture, but monoclonal antibody will bind one epitope, right? It is against a particular epitope. It may not be able to recognize, bind all the cell, all the infective cells and remove them. That's why you need, when this kind of a response comes, the immune response we develop is a polyclonal immune response, which binds to many different parts of the, so that's why it's more effective. Monoclonal response is not as effective in clearing infections. It has its other uses, but not for clearing infections. That it is, you need to have a repertoire of antibodies which should be able to bind different, because suppose organism is hiding somewhere and only this part of it is exposed, and this is not recognized by the monoclonal antibody, it will remain there. It has, and that part which the monoclonal antibody binds has to be accessible. Then only it can neutralize it. And also it should have the property of neutralization, or it should have the property of being able to remove it from the circulation. That's why the response which develops generally is a polyclonal response. So generally the T-independent antigens are polymeric antigens like carbohydrates, which result in this T-independent kind of an immune response. And there is a special class of B cells which are known as B1 cells, which do not require T cell help. They mostly produce IgM antibodies, and most of them are auto antibodies, but not very high affinity antibodies. The reason for this B1 cells and this production of IgM antibodies is they have low affinity antibodies and they bind to a lot of self antigens. So if cells are dying, you also have self antigens being generated all the time. So the function of these B1 cells and the IgM, low affinity IgM autoantibodies which are secreted by them, is like a housekeeping function. Their job, it's like garbage disposal. Basically, they bind these self antigens and remove them, so that they don't cause any problems. That is what is believed to be their function. So this is summarizing what I've been saying. And I think this is kind of my last or second last slide. That's why I said that we will start with the methods in the next lecture. And this is kind of the summary slide, that B cell activation is much more well characterized, because remember, immunology started with vaccination and B cell memory was the first thing. So a lot of work has been done on B cells. T cells also a lot, but T cell memory is still not as easy to study as B cell memory. So more is known about how B cell memory develops than T cell memory. And antibody responses, most of the organisms, you will require a T dependent, you will require T cell help. Only in certain cases the T independent antigens can help in clearance of infection. But mostly you should worry about more the T dependent immune response. And so memory in general is an area which is, I think, will continue as a research area for quite some time, because everyone wants to generate excellent kind of memory, so that which is long lasting, which is highly efficient, which is present in the place where it is required. So that is an area which is an area of active research, and I think it will continue to be in that in a near future also. So that is what that's what I am going to tell you about the T and B cells. And more will come as we study, other diseases like cancer. In each cases, we will cover more of this. Okay, thank you. I thought now we are done with this part.
I'm sure you are enjoying the lectures, starting with basics and ending with the problems. Correct? You know that immunology is an open area. As we know, as much we know, we start asking more and more questions. Though the Nobel Prize has been given to the Susumu Tonegawa for antibody diversity, I was just listening to the lecture sitting in my room when you are talking about the VJD reasons recombination, and almost similar mechanism is for the T cell receptors also. The number of diversity generated by these receptors, if we calculate the potential which we are having, the number is more than the particles present in the universe. So that capacity is with us. If we start thinking like this, then we should realize that still we have not understood 1% of the immunology. Correct? And we cannot see each and every outcome or what we are understanding in isolation. We have to correlate this with other mechanisms. It is very complex. So I'm sure at the end of this course you will find the areas in which you can plan your research, you can build your own career in immunology. Not only in immunology, maybe in the diagnostics, maybe in the developing the diagnostic tools, maybe developing the instruments, maybe using in this application in increasing the productivity of livestock. It's a completely open area, and we are taking this advantage of this GIAN program for this, that we are in this hall or in this type of program, we get the people from the different discipline in one hall and spend few time, say few days, and we come out with the ideas. So I think we will break here for lunch. Lunch will be served within a few minutes, and then we will come back around 1:30. 1:30. Thank you.
the difference between CTLA4 and PD-1 is CTL
Hello, hello. Hello.
So most of the things I'm talking about are more of an overview. So I have not gone very much in depth in most of the things. But if you want to know more in depth, you can either let me know, I can tell you, or because otherwise each topic by itself can become a lecture. So that's why first is more that you should be introduced to a subject, and then you can go in depth into whatever interests you. And so this part of this session, basically I'm going to talk about some of the techniques which you can use to study immune cells, but these techniques are not restricted for study of immune cells. These techniques can be used in general. So we will start with flow cytometry, because it is an integration of basically engineering and biology, which has led to the development of this technique. So immunology, or in fact, in the recent years, you would see that all different disciplines are now coming together, and it's more interdisciplinary approach to answering questions, which seems to be working. Tomorrow or day after, when I will talk about cancer diagnostics also, you will see how many new things are coming from biomedical engineering sides, which are being used for diagnosis, and how you can develop some different models based on biomedical engineering. So approaches. So flow cytometry is it's a technique which I would say has revolutionized immunology. It's kind of, if you want to do any kind of immunological research, it is a technique you have to learn to use. So what it is used for, and what it started to be used for, was to characterize immune cells. So if you see in the lectures, I told you that you have CD4 cells because they express this marker CD4, or you have CD8 cells which express this marker CD8. Expression of marker is fine, but you have to be able to visualize these cells. Flow cytometry allows you to do that using these special markers which are present on cells. Right? So what is flow cytometry? Flow is fluid. Anything which flows is fluid. Cyto is cell, and metry is measurement. So measurement of cells via fluidics. Basically, this flow cytometry is not a microfluidics device, but a fluidics device. And now people are trying to make miniaturized models of this. It has become smaller and small, but there are some handheld flow cytometers which can detect maybe not so many colors, but they are useful for if you need to look for one or two markers. So flow cytometry is a method for measuring multiple physical and chemical characteristics of the cells by passing a light or on a stream of suspended cells. So basically what it is you pass cells through a stream, light strikes, and there is a fluorophore. Maybe I can show you, that's more easier. So it's essentially an automated microscopic technique. Earlier people would look at the microscope and see, okay, where there's fluorescence, or where this cell is here. This is more of, there you have to look under the microscope and see the cells. Here you can, the machine does the visualization. And the other advantage is that you can detect many colors on the flow cytometry. And now things have become so advanced that you can go up to 18, 20, or even as things are improving, we can go more, even though it is not as easy to use, but you can. And it also allows you to detect rare cells. And so the other major advantage is you can even collect those cells, live cells, and you can sort. It's known as flow sorting, but you can sort out these cells, and now you have a purified population which you can use for your experiments. So it started with pulse cytophotometry when the first patent was made, but the name was changed to flow cytometry. And now most places you will see that people don't even write flow cytometry. It's mostly known as FACS, and that is fluorescence activated cell sorting. And this is a name given by the company Becton Dickinson, because they were amongst the first to come up with these flow cytometers. In fact, in the beginning, most of the labs, the cytometers were from BD, Becton Dickinson. So they seem to be synonymous with this flow cytometry. And then once the flow cytometry was developed, it started being used for clinical diagnostic tests. So for that, like, you had special cytometers, because when you use for clinical diagnosis, these instruments need to have a, at least in US, they need to have a clear certification. If they don't have that, you can't use them for clinical diagnostics. So there were certain instruments which were made by BD, which were only dedicated for this. But first, let's see what are the advantages of flow cytometry. So it can be used for characterizing and analyzing particles of different interests, such as cells, chromosome, bacteria, and even some people use it for different particles. Like nowadays, people are looking at exosomes, which are small bodies which are given off, secreted from the cells, budded off from the cells. They can also be detected by flow cytometers, but not all cytometers can detect it because those are very, very small in size. Some people even use it for detecting some kind of marine organisms and things like that. So, but one of the few things which you have to remember in flow cytometry is cells have to be in some kind of a suspension because they have to pass through. It's not like a microscope where things can be fixed. So anytime, even if you have a tissue, you need to make a suspension of cells from it if you want to use flow cytometry. Okay, this is not as clear, but basically there are many, many different uses of flow cytometry. You can measure cell cycles, cell proliferation, even cell death, apoptosis you can measure. And then, so this I will show you later, that how you can even differentiate the cells based on size, granularity, how just that can also, you can differentiate the cells based on these functions also, or properties of the cells also. Then you can look at things which are intracellular, if they're producing a cytokine, if they're expressing a transcription factor, or some signaling molecule is there, that also you can use flow cytometry to detect. So not only can it detect cell surface molecules, you can also use it to detect intracellular events. And this is another form of signal transduction where you're looking at basically calcium signal, because whenever all these lymphocytes get activated, there is a calcium flux which occurs, which you can measure by flow cytometry. So depending on the antibodies you use, you can find out different kind of cell type because depends on the marker. So you can differentiate between hematopoietic, lymphoid, non-hematopoietic cells. You can have different cell lineages. Is it a T cell or a B cell? And also you can look at the activation stage of cells, because in the morning only I mentioned that like dendritic cells, when they become activated, they become ready to present antigen. So now you can look if the HLA-DR, which is the MHC molecule, has it gone up, the expression of CD80, 86, which is the co-stimulatory molecule, does it go up? If it's higher than what you saw at the baseline, then those cells are activated. So this is just an example to show that the sizes of various kind of cells. So bacteria are so small, 0.5 microns. Then phytoplankton is a little bigger, red blood cell a little more bigger. These are beads from BD, which are usually used to set up the instruments and things like that. And then you have the lymphocytes, neutrophils, monocytes, and you can go larger. And cells, when they become activated, they can be larger than what it is shown here. Usually they become larger than when they are unactivated. These are just some examples of how the flow cytometers have looked over the years. This is the most common of the flow cytometer, which you will find in most labs. This can detect, it has 2 lasers, so it can detect up to 4 colors. Then came Canto. I think it also can detect, it has 1 more laser, so it can detect 8 colors. And then LSR2 came, and now there's BD Fax Area. And now, in fact, a lot of companies have made flow cytometers, which have different properties, which in some case you don't need compensation, but now you can easily go up to 14, 15 color for sure, where it works, even though some people say you can go higher, but then it becomes more and more difficult. So flow cytometer basically has three major components. Fluidics, where the cells will go. Then you need to have an optical component where the light should strike those cells. And then you have to have the electronics component where the signal which is generated should give you basically is converted into a graph so that you understand really what is happening. So in all flow cytometers, you have a sample tube, and there is basically this kind of a thing, sheath it is known as, and cells are sucked up in a straight line via it. Once they reach a flow cell, a laser will strike, and so okay, I'll move to the next and then I will tell you. Once the laser strikes, basically before that what you are doing is you are taking your cell, you are adding an antibody which is labeled with a fluorochrome. So when a laser strikes, a fluorochrome, what it does is, once the laser strikes, it's going to emit light at a certain wavelength, and that is what is really detected by the electronics and converted into a graph. So this is all more for engineers, is what I would say, that when a particle passes through the laser, interrogation point, which was shown in the last one, any fluorescent molecule present on the surface or on the particle will fluoresce. Because laser is striking, there is a fluorescent molecule, it will become activated and it will emit light. And then there are lenses are there which can collect this scattered light, and then this collection components are then go through detectors which basically give an output for us in the form of a graph. So this it lights into electronic signals, digitizes data, and this computer basically gives you a kind of a graph which shows each cell as a particle. That is kind of the basis of it. But so even if you don't have fluorescence attached, first, if a light is going to strike a spherical or any kind of a molecule, it's going to scatter, because there is a solid object there, it has to go, it cannot pass through, it goes this way or that way. So there is forward scatter and side scatter. This will change with the size of the cell, because if it's a small cell, scattering is less. If it's a bigger cell, scattering is more, right? Side scatter, the same thing. If it is more granular, more scattering, because there's more things to strike. So there's going to be more scattering. So based on this is not very clear, so I'll show you in the next slide. So you see, based on the forward and the side scatter, these are basically, this is whole blood. Only doesn't have the RBC because lysis has been done. So that's why you see so less number of RBCs here. But these are all lymphocytes because these are smaller cells. Light scattering is less. These are monocytes, which are kind of intermediate cells. And this whole big population is the neutrophils or granulocytes, because the maximum scattering of light is via granulocytes and this. So that's why they have come up. Both the forward scatter and the side scatter is much higher in these cells. So you can even distinguish using flow cytometry without any kind of fluorescence molecule based on size. But this is usually not considered very correct, because sometimes when monocytes become highly activated, they may fall here. Similarly, if the lymphocytes are too activated, they may merge in this area. So this is not a very correct way of delineating cells. But if you are looking at whole blood, you can start with looking at that particular area. So that's why generally only forward side scatter is not used. It's always, that is generated irrespective of having a fluorescent molecule or not. It is always interpreted in conjunction with flow cytometry, flow data. But it is a good beginning, like, okay, we know that we want to look at neutrophils, let's look at these cells first and then worry about what is happening here. It's a good starting point. So you have various fluorochromes because you need to have fluorochromes of which will get excited at different wavelengths and which will emit light at different wavelengths, because if all of them are going to do the same, you won't be able to study the different cell types. So one of the things which happens in the what you require for flow cytometry is you need antibodies that are conjugated with fluorochromes, because antibodies are molecules which are against a particular cell type, particular molecule protein. So if you need to look at CD4 cells, you need an antibody which is against CD4, but it should have a fluorochrome attached to it, because just the antibody binding, if it is binding to CD4, you have no way of visualizing it. If it has a fluorochrome, then the machine can see it, because this is what I mentioned. So these are very common fluorophores, which FITC, which is green colored signal. This is usually red, and then there's APC. These are the four most common ones, but now there are so many different kind of dyes which have come, which excite at different wavelengths. And many of these fluorochromes have come from the study of, in the deep sea. If you go down at a certain depth, most of the creatures are highly fluorescent. And from those, a lot of dyes have been developed, because like DS red, and these dyes are more stable also. So, and these creatures are fluorescent. So what I'm trying to say is so many things from different places can result in, these are mostly organic compounds, but the newer dyes have come from the study of the deep sea creatures. So fluorochromes, basically this is just to show you a molecule known as, the green signal which I was telling you. There is excitation at this wavelength and then it emits at this. And then there is a diode which will capture that and you can see that. So a receptor is expressed on the cell, okay? It depends. It may be expressed at very high levels. It may be expressed in some ways that there are low to high levels, or it may be expressed at a much lower level. So all these things can be looked at by flow cytometry. Here is an example. So you have the receptor CD4, because this receptor is expressed at very high levels on your T cells. So if you do a staining, you get this kind of a population in the blood, that these are all positive for CD4 and these are cells which are not positive for CD4. So there is a very clear demarcation if you see. But then there are also molecules like CD45RA, where some cells are expressing it at a medium level, which you are seeing here, and some cells are expressing it at a higher level. So then you need to use basically another marker to see which are the cells which are expressing it at an intermediate level, which are the cells which are expressing it at a higher level. So you can also do this kind of an analysis. Then there is this molecule which is expressed at a much lower level. It's not going into this far region. So it depends on the density of the receptor on our surface, that how well it is expressed, so it's always easiest to study these kind of molecules because they show a very high level of expression. Followed by this, and these ones are the most difficult to study. So this is what happens. Basically, you have a fluorescent signal. Cells are labeled with these antibodies, and fluorescent this is what I was mentioning. The intensity is directly proportional to how much antibody with the FITC. In this case, you have much less. Your signal is going to be much less. The density of the receptor, you have two only receptors. You have many more receptors here, so many more antibody binding. So you are seeing you will see a much better signal. So it kind of also tells you in a way that to what level a receptor is expressed on a cell. So when you are choosing these dyes, fluorescent dyes, you have to be careful because sometimes the spectra overlaps. So here is an example that you take this green dye and this red dye. So there is a part where these two overlap. So if you are using these two dyes, and you may get a false positive. Like suppose you have only FITC there, it's leaking there in PE, so you will see a signal there also. So and you will think that the cells are double positive. In reality, they are not, because it's simply because of leakage. So that's why you have to do something known as compensation. So either you should choose dyes which are far apart, which have no spectral overlap, if you are doing multicolor staining, or you have to make sure that you have to change the voltage and other things so that now this overlap is not there. And otherwise, you have to use many controls, in the sense like you have to have single colored cells stains, that okay, when you have only FITC, you are seeing this much of a staining. When you have FITC only PE, it's here. When you have both, is it lying? You have to set the gate that above this is what I will, this is the threshold, and above this I'll consider positive. So those things will only come with experience. And the problem is, I'm talking about 2 dyes. Now when you talk about 13, 15 dyes, it becomes more and more difficult. And major problem is then you have to have, for 2 tubes to, you want to look at 2 kind of cells, and you've done 13 color staining, you end up with 13 tubes only for controls, and 2 tubes which are basically your experimental tubes. So to really find out that your data is valid, you need to have so many controls for that. So as you make your flow cytometers more complex, it also requires that you need to be more and more careful that your signals are really what, because it doesn't look at the cell, it just looks at what the fluorochrome is.
Software is not there to.
Software is not able to compensate for it. Software is not there for that. You have to have, software will do if you have used the control tubes, because software uses that, or you also use the same thing, it uses it to set a threshold. It needs some kind of a reference point, because software cannot make a reference point, right? It can only know, okay, when you have these 2 dyes, it comes here. That is, and each cell also gives a background fluorescence. So sometimes that also changes the the way cell is activated. Sometimes even one particular thing will bind more. So you need to have a particular cell control. You cannot, like if you are using lymphocyte, you need to have lymphocyte controls. You cannot use a monocyte control. So that's why you need to do controls with each and every experiment and that's the reason there are some programs which nowadays tell you that you can change the compensation afterwards if you find that what you have acquired has a problem. It's not an easy thing, but it's not a simple program. So now we come to most of the uses of flow cytometry. Here, some of the things I'm mentioning are for clinical immunology, but it's not necessary. You can use it for things in the lab also. So first thing which the flow cytometry is most used for is phenotyping. Phenotyping is looking at the receptors on the cells and finding out if they are activated, unactivated, what kind of a subset it is. So basically characterizing a cell. And then the other thing which you use the flow cytometry is to look at the functions of these cells. Are they dividing? So that is proliferation. If they are cytotoxic, how do you look at if they are cytotoxic? Should they express a molecule? What else can you do to confirm that? You can also look at phagocytosis. You can look at other functions like reactive oxygen species and other things by flow cytometry. So first thing is usually in clinical immunology, generally there are thresholds, like people know that number of lymphocytes in this population, in the Indian population, should be say from 10 to 6 to 10 to 8, or that is the range. So if you are below or above that, then there's something abnormal. There's always a range because these things are never fixed. So suppose you find that in your, you do a flow cytometry and you find that the number of B cells in a general population are about 5%. But in this subject, it is more than 25%. So why are these B cells there? That means there's some abnormality. Half of the time in case of B cell related cancers, it's because one clone has proliferated so much that you are seeing so many B cells. Similarly, if T cells are overactivated, it suggests maybe there's some infection or something ongoing. But just by the numbers also you can find out that, okay, if it is not within the normal range, then there can be a problem. If you have a missing CD4, when you do this enumeration, you find that the lymphocyte, especially the T lymphocyte number, is very low. Then you start suspecting things like, could it be HIV? Could it be, then you do further, then you look at CD4 cells, then you look at function, you do an antibody test. So it gives you a clue that there is some kind of a problem there. So these are just, some examples where you find differences in the number of T cells. So there's patients which have primary T cell deficiency, Wiskott-Aldrich syndrome. So in this case, the patients do not make a T cell response, and T cells level continue to decrease with age. So if you find, as I said, if you see that your T cells are very less and the patient is suffering from infections, you have 3 to 4 choices. Okay, these are the possible choices. Then you go further and narrow it down. Same is another syndrome, New-George anomaly. In this case, you have also decreased number of, because the development in the thymus by itself is not normal, so the cell number is decreased. So in both these cases, you have T cell deficiency, but the reasons are different. So then you go more deeper into finding out what it could be, this or this, and then you look at the property and decide, okay, do further tests and decide it is this and not this kind of thing. And similarly in AIDS, which I already mentioned, that you have less number of CD4 cells. So this is another lymphocyte related disease where you have autoimmune lymphoproliferative syndrome, ALPS, and in this case you have large number of lymphocytes. So they're basically dividing unnaturally, right? So in this, sometimes you will see that doctors touch your lymph nodes or this area and these are swollen. It also usually indicates infection because cells are proliferating, and that could also that will lead to enlargement. So some of these things you can, if it is an infection, it should subside. The lymph nodes, once the infection goes away.
Migrate?
No, lymph nodes will not migrate. Lymph nodes are fixed in places in your body. The cells can migrate to lymph nodes.
Yes, if it is overloaded, then it can migrate.
Lymph node will not migrate. Cells from the lymph node can migrate, but not lymph node itself. So basically, this is just to show you that so many different kind of things can be there where you have problems in T cells, and all these can be you can use flow cytometry to look at all these different conditions. So this is just to mention that there is an enzyme known as BTK, which is also, so most of the diseases which are linked with any kind of lymphocyte deficiency are known as primary immunodeficiency, because if your T cells and B cells, either of the cells are not functional, you are not going to make an adaptive immune response. NK cells and cytotoxic T cells, I tell you that they can lyse cells, right? When cells die, what happens is now their DNA is becoming exposed. So if you use a dye like PI, propydium iodide or 7-AAD, what happens is it now can go inside the cell and bind to the DNA.
You are saying no, BTK gene mutation causes the immaturation of B cell. B cell will not mature if mutation is BTK. So you said that they are doing gene therapy and to cure the disease.
I don't think it has. People are trying it, is what I'm saying.
Because one of my colleague cousin, they have this problem. So the doctor told the same thing. It's gene therapy. But how far it will work, we don't know. Same thing. Every 3 weeks when they go and then take this exogenous antibody injection and come. So almost 14,000 they're spending every 3 weeks once.
See, that is one of the problems with immunotherapy, that so far, because all these are biologicals, they seem to cost a lot more than regular drugs, chemical drugs is what I would say. I think we have to maybe the field is still new, but we have to learn to develop techniques to make the therapy a little less expensive.
Again, ma'am, growing those, suppose if we are doing stem cell therapy, we have to take the...
That's what I'm saying, that because of this, it's much more expensive. But maybe we have to devise ways that we can grow them maybe more efficiently, using less things and less manpower or less facilities or less... basically, we have to try to make the process less expensive. If we need to use it for general public, or if the general public...
At least for this disease, only one gene got mutated. So we can, if you replace with an intact gene, then it can be solved.
It can be solved.
Cancer.
Yeah. Again, it's... So basically, if you want to see if NK cell are killing something, you basically label the cells with the dye. It's the CFSC dye. Basically it makes the cells green. It is there when cells are live, and the 7-AAD will only stain the cells when the cells are dead. So here, if you are looking at cell death, these percentage of cells are dying. So that means if there's no cells here, because these are all 7-AAD expressing cells, because this axis is 7-AAD, this axis is CFSC. So these cells are not expressing CFSC, or they are basically double positive. CFSC expressing, but 7-AAD also positive. So these are dead cells. So if you see dead cells, that means NK cells. So this is one method which you use. It has to be, I will have to explain more in details. Basically, you have to do NK cell and targets, whichever cells you want to see killing, you have to incubate them. Your target cells are labeled with CFSC, and then after, it takes about 4 hours for this assay. In that, you have to do different ratios. You take 1 to 25, 1 NK cell to 25 target cells, and this way, and then you look after 4 hours, you stain the cells with 7-AAD and see how many cells are dead. So it tells you, and it should be proportional. If you are increasing the number of NK cells and the target cell is remaining the same, you should see more and more cell death if your NK cells are killing, right? Because if you have 1 NK cell and you have 50 target, you should see some cell death. If you are now making, instead of 1 NK cell, you're making it 10 NK cell, and still the target is 50, you should see more cell death if it is bigger. So that is one way you can measure. Earlier, people used to do this using chromium labeling. So many of the radioactive based assays are now people are trying to replace them with other assays, because there's a lot of radioactive waste generated if you use those assays. And radioactivity, as you know, can remain for a long time. No, same sample. Like...
Is it here going for the cells which are live?
Cells are live, yes.
And which are going under the apoptosis process. So that for that purposes, we have to use the same sample.
Same sample. Basically, you take the NK cell, you take your target cell, which can be any tumor cell or something, which you have labeled with CFSC. You incubate them for 4 hours, and you expect that your NK cells would have killed some of the tumor cells. And then you add this dye, 7-AAD, because it can go inside only dead cells, it will stain those dead cells. And then you acquire on the flow cytometer to see how many of those cells, percentage of what, like starting cells are 50 cells, if it is, those are 100% cells. Out of those, how many are AAD positive? So that is your lysis factor in a way. You can calculate. So So you can use flow cytometry to characterize, different patient cells. So here is an example. This is, so in the division I am in, it's the division of basic and clinical immunology, and there are many physicians. So there is a whole clinic which is dedicated to primary immunodeficient patients which have defects in T and B cells. So some of these patients which come, if they show some certain characteristics which are kind of unknown, then we try to look at their lymphocytes and make basically a research paper out of it to see what kind of abnormalities they have. So this is one of the patient where there's colitis and in primary hypogammaglobulinemia. So when basically hypogammaglobulinemia means there is less number of immunoglobulins in the circulation of these patients. So they are deficient in IgG. So with normal CD4 T cells deficient, but what I wanted to show here is this is control and this is patient. You are looking at different markers. These are all CD8 cells, okay? And you are looking at, so in many times what you do is as a control you use an antibody which is exactly of the same isotype. Mostly you use IgG. If it's IgG2A, you use IgG2A. It also has the fluorochrome because it gives you, if that kind of an antibody is added, where it binds. It's a non-specific kind of a thing. So that's your background. We call it isotype control. Okay. Now you use an antibody of the same kind, but it is against granzyme B. So it should bind more than this. Then only we will consider it as positive. Basically, you use this to set this gate that till here, if this non-specific one is binding, this is all non-specific. So you use, basically it's a control, you can say, where this is your baseline. You have to have a signal above that to consider it to be considered positive. So I told you that the CD8 T cells, the way they kill is they have to have cytotoxic this granzyme B molecule, and CD107 is basically a molecule which is also expressed on cytotoxic T cells. It is a measure of when these cells become cytotoxic, this molecule is supposed to go up. It's also one of the markers which is supposed to go up on these cells. So that's why we chose these two. And here, as you can see in the control, you have granzyme positive cells, but you don't have too much of CD107 positive cells. And here we are looking at perforin, which is the other molecule which has, so this is expressed. But if you look at the patient, the number of granzyme positive cells is much higher. Similarly, the perforin expressing cells is also very high. क्या हो? So what it shows you is that the patient CD8 cells are much more activated. They seem to be much more highly cytotoxic. So too much of anything is bad. So it could be. So basically, if you see anything different, and this is baseline, you've just taken blood from these subjects, there's no reason why it should be activated, why these CD8 T cells should be activated in these patients, but they are showing a difference. So if you see too much of an activation of CD8 T cells, it means they may be killing other cells because they are activated. So that can lead to problems. So it is proportional in the sense when, if you're seeing granzyme without perforin, it will not work. So it should, but always people want to see both. That it's more a question of, okay, you're showing granzyme, if it does not have perforin, then what? You haven't shown it. So that's why whenever you do, you try to do both. That is like one of the reasons. And in fact, nowadays people want, like, you are showing it with one thing, maybe you should have another to confirm it. It's more a question of that than So this is another patient who had this Good syndrome, and they also had leukemia. So here we are looking at in these patients these different markers: PD1, granzyme B, perforin, CD1. So mostly we are looking at CD8 T cells which are there. So these two are control panels, and these are patient. And this side is all isotype, which I told you that it's basically the same kind of antibody. If it's an IgG1, it will be an IgG1 coupled to the same fluorochrome. But in this case, it will not bind, it's not supposed to bind to granzyme B. So this is your background. But here you add the same antibody, the IgG1, but it is specific to granzyme. So that is the difference. Okay. So if you see here, control cells are expressing low levels of PD1, granzyme B is hardly there, perforin is hardly there, and CD107. So these three molecules are molecules of activation, and this is the inhibitory molecule. PD1 is what I showed you, or the inhibitory co-stimulatory molecules. It's expressed at much lower levels. But if you look at the patient, so we do isotype for patients also, because I told you that sometimes some cells, some antibodies have a tendency to stick. So in patient, we always want to set a background based on that kind of staining. So if you see here, it is a little higher compared to like this one. It's a little higher. But this is just to show that isotypes can vary with, it need not be that, okay, once you have set the isotype, it's going to be, you should do it each and every time. But in patient, again, you see that they show high levels of granzyme, hardly any perforin, and very high levels of PD1 also. So what does it mean? It has activatory signals also showing granzyme, but granzyme may not work as well because there's no perforin, and it also has high levels of the inhibitory co-stimulatory molecules PD1. So this is kind of an abnormal kind of a behavior of T cell. So maybe this is why the T cells in this patient are not functional. So this is basically, these examples are just to show how you can characterize this patient population. And maybe, so once you know, okay, these CD8 T cells are not behaving properly, they have these abnormalities. Now you can maybe do other assays to see what is wrong with these T cells, why are they behaving in this way. And many a times people have uncovered mutations or some kind of a genetic change or some reason why this has happened. So then it becomes a very nice science and nature paper. So there are patients where you have very high levels of IgM, and these are known as hyper IgM patients, and they don't have much of IgG. So what it tells you is, in these patients, if there's so much of IgM, and if you remember my B cell lecture, IgM is the antibody which is secreted early, and then it has to class switch to IgG. If you are seeing more of IgM and less of IgG, that means there's some defect in class switching, right? Class switch is not occurring. Is it because the B cell itself is unable to class switch, or is the T cell help not enough for it to be able to provide like help for class switching? Where is the defect basically? But so some of the defects have been identified. One is that the CD40 ligand interaction is required between T and B cells for the B cells to proliferate and class switch. So it has been shown that this molecule is either deficient on the B cells or the antigen presenting cells, so this is not happening. Sometimes the ligand on the CD4 T cells is not there, so again the interaction will not take place. So the defect can be on B cells where you don't have expression of CD40, or it can be on T cells where the ligand itself is not expressed. The other thing is T cells are not getting activated at all. So then they are unable to provide any kind of T cell help because they are not activated. So there are many class switch memory defect. So there can be also the T follicular helper cells are not there, so there is no class switching of memory. So there are many, the enzyme AID which I mentioned is required for somatic hypermutation and class switching, it is deficient. So there can be many reasons. So first thing is you find out if you have cells which are changed, then you go for different mechanisms which are there. So interferon signaling is defected in certain bacterial pathogens like mycobacteria. So you can look at the receptor expression for interferon gamma. If it is down, then even though interferon gamma is being produced, it won't be able to act because the cells are not expressing the receptor for that cytokine. So cytokine is not going to be able to function. So all these examples are just to show you that how much of a use flow cytometry has become more or less essential in characterizing, cells in patients. Is mostly in the blood. Now some analysis is being done in quite a bit of analysis is being done in tumors and other tissues, but only if it is removed by surgical or some other means. Otherwise, blood is the most easily accessible thing which you can get from humans. You really can't get much of other things. Some of the biopsies are available, but the problem is the size of the biopsies is so small that you don't get enough cells to perform flow cytometry sometimes. The other problem is also that the hospital should be associated with a lab so that the moment biopsy is taken, you cannot keep it for very long. The cells will start to die. Otherwise, you will fix it for for the sections and other things, pathology, where you add it to formalin. Then you can't do flow cytometry. You need to have live cells. So that is why the blood is the most common thing. Blood, you can even transport it overnight. Then also few cells die, but you can still do flow cytometry. So in this case, this is just to show you that this patient had basically selective IgM. So in this patient, we did not see any IgM antibody. We see only IgG. It's the opposite of what you see in hyper IgM. So now the question is, if the IgM is not there, where is the IgG coming from? Why is the IgM not visible? Is it not being made? If there's nothing there in IgM, how are you having class switch? So we are still studying that patient, but here is just to show that we looked at two different, and this patient had high levels of this cutaneous Mycobacterium avium infection. So interferon gamma is involved in response to Mycobacterium. So that's why we looked at the expression of this on both lymphocytes and monocytes, and these are controls. So both, in this case, on the lymphocytes, the expression is little more or less similar. But in this case, in the monocytes, it's lower. If you look at the mean fluorescent intensity, if you calculate the mean, there are programs which do that. It's 155 and it's 226 in the control. It's not like a major deficiency, but still it seems to be sufficient to cause. And then we looked also at another receptor, IL-12 receptor, because IL-12, I told you, when it is secreted by antigen presenting cells, it acts on T cells to induce interferon gamma. So that's why that was also looked at, but not a major difference. Mostly the difference was in interferon gamma. But we are still, as I said, studying this patient because what we've realized is they seem to make the IgM, but IgM is not secreted out. It seems to remain inside the cell. So it could be simply a defect that it is not secreted out. Maybe there are some signals which are required. So now we are trying to see what kind of signals are required which make the IgM to be secreted out. But still, we have not answered that question. What I'm simply trying to say is that each patient is presents a problem which now you have to work out why this has happened. So it's like an experiment of nature. Now you have the phenotype there. Now you start to think, why is it happening? So you already have the final outcome. Now you need to work out why is it happening. So in chronic granulomatous disease or CGD, in this case, neutrophils don't make a reactive oxygen species. So again, these patients are more susceptible to infections. So, but you can use flow to test this also. So procedure is also written, but basically what you do is you load the cells with this dye, DSFDA or DHR, and when you activate the cells, the reactive oxygen species, if it is formed, it makes this dye fluorescent. Otherwise, the dye is not fluorescent. And then you can look at it by flow cytometry. So the example is it's shown here. So this is what happens. You load the cells with this, they will not fluoresce. But if the oxidation and everything is happening, then they will fluoresce and give a green kind of fluorescence, which you can see under the microscope also, or you can look at the monocytes and macrophages, monocytes and neutrophils by flow cytometry. It's shown here, we can look at. So this is a control subject. Resting, this is what it is. When you have stimulated, you see this high levels of fluorescence. The whole cell population has shifted here because the cells are highly functional and they're making this reactive oxygen species. But if you look at CGD patient, there's no shift. So when you see this kind of a thing, then you will start to think that there is a problem with the reactive oxygen species in this subject. And this is just to show this is a carrier. So if it is a genetic disease, then if you have half from one parent and half from other, so you're half functional, then you're seeing this kind of a pattern. So it's not a fully functional neutrophil. It is still able to make ROS, but not to the level of the control. So you can even tell that, okay, in some patients the ROS is being made, but not to the extent it is required. That could also be, it's partial deficiency. You can also look at phagocytosis because you can label the bacteria with this dye. And now if it is uptaken by the cells will become fluorescent. If they are not uptaken, the cells will not be fluorescent. So this is used very regularly. Sometimes people even use just beads, polystyrene beads coated with this dye. So SCID is basically SCID mice. You may have heard in SCID what you have severe combined immunodeficiency. So in this case, both the T cells and B cells are missing, either missing or not functional. So there are different kinds of SCID, but basically, ultimately, in the T and B cells, either not missing or not functional. And you can look at this by flow cytometry again, because if there are no cells and B cells, you will not get any staining. So these are some primary B cell deficiencies. B cells are X-linked. So if you're really interested, basically in all these cases, first thing is when you look at first thing the doctor does is he looks at the immunoglobulin level in your serum. If it is deficient, then he starts looking at these B cells, T cells and other things, because if it is changed, then there has to be some problem with any one of these lymphocyte subsets. And these are all different forms of primary immunodeficiencies which are there, which are more for people who are really into that field. And so they now fall into this phase. This is CVID, common variable immunodeficiency. So these very different kind of immunodeficiencies are there, and those are known as all CVID now. And you can do so many different, you can do all this different tests. So here is just to show you that we are looking at different kind of B cell subsets. There's IgM memory. We are looking at marginal zones. These are class switch memory, naive B cells with different markers. So in the patient, if you see, there is an increased percentage of this transition B cells. You also see increased number of these IgM memory B cells. So basically, we have these panels of B cell antibodies which will tell you what different kind of B cells are there. We have panels of antibodies which will tell you different kind of T cells. And if a patient comes and it shows some abnormality, we do this kind of a thing to see which particular cell population is missing or changed, increased, decreased. If there is some change, then we go further. So this is just to show an example that how many different kind of B cells you can look at by flow cytometry. Class switch memory. We have marginal zone. You have unactivated B cells. IgM memory. this is transition B cells, plasma cells. All these you can look at by using a combination of just 4 or 5 markers. So for T cell activation, generally when T cells get activated, these are the 2 molecules which go up. CD25 is basically the IL-2 receptor chain. And it goes up because, as I mentioned, the T cells, when they become activated, they will secrete IL-2, and the IL-2 then acts itself like in an autocrine manner on T cells, and it causes their proliferation. That's why CD25 goes up, because it needs to now bind to IL-2. CD69 is also another molecule which allows proliferation. So they have functional values, but you can also, because they become upregulated on activation, you can use them as activation markers. That if the level is here, if they are showing like 10 to 2 in the plot, they will now become 10 to 3. The density of the expression of this receptor will go up, so you will see more binding, so more fluorescence. Okay, so you can look at T regulatory cells, T helper cells. You can look at the transcription factors which are present in these cells to look for these, to differentiate these cells. And this I'll show you now and later. Basically, here I'm just enumerating what different things you can do. So T cells interleukin production. So I was telling you about intracellular, how you can go. So far whatever I have talked about is, you have an antibody and you have a cell surface molecule, you are allowing it to bind, and you are using flow cytometry to detect. How are you going to look at intracellular molecules? Now the antibody needs to go inside the cell to bind. And also if you're looking at cytokines or immunoglobulins, these are being secreted out. There may not be enough concentration inside the cell for you to detect. So in that case, what you do is you first use you fix the cell and you use a kind of a very mild detergent, which removes lipids from the bilayer. Basically, it allows the antibody to go in. So, but it is not enough to kill the cell. Its cell still kind of retains its shape and function. Not function because you're fixing the cell, but it still is like a cell. It hasn't become totally deformed. And also side by side, you add something known as Golgi stop, or basically something which prevents secretion of the molecules out. So now they accumulate inside the cell. They are being made, they will accumulate, they cannot be secreted out. So then when the concentration is enough, you are able, the antibody you add will bind, and now you can use it to see by flow cytometry. So you can look at proliferation of T cells also, and this is shown here. This is a dye which is known as CFSC. So in this case, what you do is you take the cells and you load this dye, and this is a dye which has been made in such a way that it doesn't kill the cells. You just have to incubate the dye with the cells for 10-15 minutes, and it goes inside, and now all your cells are green. In fact, if you look at the pellet, it does look a little green. So when these cells are dividing, the dye is also dividing with it, right? So if you're starting with this X concentration of dye, here, cells are highly fluorescent because you have just labeled them with the dye. If they are dividing, the dye is becoming less and less because it's going into 2 daughter cells. The 2 is going into 4 daughter cells. So you can see proliferation with this. In fact, you can see how many divisions it has taken place, because this is where you started. This is maybe 1 peak, this is 2nd peak, this is 3rd peak, 4th peak. So these many cycles of cell division have occurred. So this is used very much in immunology to look at proliferation, because sometimes you see that some cells either will not proliferate. You don't see this at all, or sometimes they just stop here, and in the control it is going on and on. So this technique is very, very much used in not even in immunology, but you can also use this. This is for lymphocyte, where it shows very nice peaks, but people have started to use it for now epithelial cells, other cells, whichever cells proliferate. because the basic principle is the same, right? So this is another patient which came in with having recurrent infections, and our physician suspected that there's something wrong with the innate immune system. So we looked at the toll-like receptor expression. And since he was suffering more from viral infections, we looked at the intracellular toll-like receptors. So you can see this is TLR3, 7 and 9, which were TLR3 and 7 are supposed to recognize the viral nucleic acids, and TLR9 more the bacterial nucleic acids. But it can also do viral. And this is the subject. So you can see this, there is some defect, but the major defect seems to be in TLR7, where the expression is nearly half, and also in this, it is highly reduced. And we also looked at the expression of extracellular, Basically, we did a whole panel of toll-like receptors. So 2, in fact, is in the subject seems to be overexpressed for whatever reason. But it's not overexpressed. If you see, so the blue is isotype control, I should mention. Blue is always the isotype control. In this, there is no difference if you look at this one and this one. The only difference is the isotype control in here is not binding as much in the patient. We don't know why. Because patient comes one time, sometimes we don't have enough, once we do the experiment, we find out that there is some problem. Unless the patient is coming again, we cannot follow it up. Similarly, the TLR4 expression seems to be a little higher on the subject. So the intracellular ones, the viral ones were down, while TLR4 is high. 5 is again down in the patient subject. So it's showing a pattern which is different from the control in certain toll-like receptors. And maybe that is the reason you are seeing this patient is showing signs of recurrent infections. So these are, I think, various examples. So this is another integrin. If the expression of this is low, then it promotes adhesion. So if there's a decreased expression, then the cells are not able to move very well. So again, they are unable to reach the site of infection. So that's why, again, these people suffer from infections. Okay, I think we will leave this. It's basically what I talked about, CGD and then LAD. So you can do it directly from the blood, this, for patients, but RBCs are a major problem. So you have to always lyse the RBCs. And RBC lysis doesn't work that easily, even though you have 100 microliters of blood, sometimes quite a bit of them remain. So many a times we do this flow, this PBMC separation using density gradient, where you take out, where you do this phycol layering, and then it removes all the RBCs. It also removes most of the granulocytes or neutrophils. If you're wanting to look at the lymphocyte population and the monocyte population, all of those are mononuclear cells. So you're basically getting rid of most of the contaminants, because in the blood, the number of neutrophils is very high, the RBC is very high. So once you have removed those, it's easier to study the population which is left over. That's why people do phycol. So here, what I'd shown you earlier, you are looking at the FSC, SSC, and these should be your, since this was whole blood, you see this whole big granulocyte population, and this is supposed to be the lymphocyte. So this is where you want to look at your lymphocyte population. So you make a gate here. So this is analysis what we are showing you. So if you want to look at only these cells, you can make a gate here, and the program will show you only those cells, and you can look at the markers on those cells. So basically, you can look at all the cells also if you wish. It doesn't matter. You can select the cells you want to look at. You can also select the cells based on, you mark these cells, or you take all these cells and you look for CD3, because that's only expressed on T cells, and you gate on these, select those cells. That all these cells I consider T cells, and I want to look if this activation marker is present on that cell or not. So the analysis of this, when you're doing the experiment, you don't worry about any of these things. The cytometer is also acquiring all the cells, but during analysis, you can go finer and finer, because if you have 10 molecules, you can look at if this is CD4 positive, CD8 negative, is it double positive, is it CD4 positive, CD65 positive. What I'm trying to say is you can make you can make subsets from just one tube. So mostly you get graphs like this, that in all, these are all T cells, CD3 positive, because they are expressing high levels of CD3. These cells, there is no staining for CD3. And out of these, only a percentage is showing expression of CD4. So rest of these CD3 T cells, these are still T cells, but they are not CD4 positive. So either they are going to be CD8 positive, they may be NKT cells, but this is just to show you that how you can analyze. So mostly it's the thing which comes in this quadrant is negative for all markers. These cells are not expressing, these are not T cells. These are the ones which are T cells. And these are the ones which are both really CD4 T cells. Low level expression of many of the molecules is there, but in other cells also. So this is another patient. Basically what we are looking at, central memory and effector memory, and this is known as T effector memory RA positive. Basically this stemra is a population which is there and generally not too much present. It's when it's present, it is considered, it's mostly non-functional kind of cells which are there. So the reason it is known as RA positive is, here is, if you have a T cell, take CD4 or CD8, and you use an antibody against CD45RA, CD45RA is a marker for naive cells. If the cells are expressing CD45RA, then the cells are naive. If the cells are expressing CD45RO, which is another marker, then cells are memory. So if you use a combination of these three, CD8, CCR7 and CD45RA, this combination, you can divide the cells into central memory, effector memory, T effector memory, RA positive. Normally, as I said, the memory should not be RA positive. It is RA negative, RO positive. But this is all dysfunctional cells which somehow start expressing RA. So in the control, you see most of the cells are CD45RA positive here and CCR7 positive. And these cells are supposed to be CCR7 positive. You always use one or two markers. But in case of patient, you don't have any naive cells, hardly any. All of them are coming in this quadrant. They are all 45 positive with RA, like memory cells with RA positive. And these cells I mentioned are generally non-functional, dysfunctional cells. So all the examples I've given you are real patients, and what it shows you is how you can characterize different patients using flow cytometry. You can even use flow cytometry for cell cycle analysis. So this is like you can look at G1 phase, S phase, and this also by flow cytometry. Like, so this is your G2M phase. The cells are in this area, then it is S phase, and this is G0 to G1 phase. So we have seen that you can phenotype the cells, you can do intracellular staining, you can look at reactive oxygen species, you can look at proliferation, you can look at cell cycle. All these things you can do with just one instrument and one cell type. So we can leave this. Basically, you can use it for diagnosis in cancers is what the whole slide is about. And this is just to show how, if you want to look at for a B-cell lymphoma, what kind of a staining technique you should use. So in some of the clinical situations, there are guidelines that if you want to use this for diagnosis, you have to do this kind of a staining. And then only you can arrive at that conclusion. There's panels of antibodies. So you have to show that. Otherwise, they will not. That's why I said that in clinical situations, many things are very fixed, that you have to do it this way. Because The problem is they need to compare it to the control, where they have set a standard based on that. CD, I think I've already mentioned that it is the cluster of differentiation, what they call. And the CD numbers are already, I think they have reached in the 400s something. Every year, whenever they meet, there are new CDs added. So that, I think, and so many markers are expressed on so many different cells that you can never remember. So for that, you always need to consult the chart or the book to decide, okay, you want to look at this subset, what does it express, what you should use.
Flow cytometry not effective in granulomatous diseases?
No, it can measure because in CGD what happens is the reactive oxygen species is not, the neutrophils do not make the reactive oxygen species, right? So you, if your neutrophils are defective, flow cytometry will not will show you that these neutrophils are defective because you are loading them with this dye, the DH, whatever dye, and if they are making the doing this reactive oxygen species, then the dye will fluoresce. So in patients, the dye never fluoresces. So then that way you can use it for diagnosis. So these are all major known T cell markers. So if somebody is planning to do, then you can look at these different things. Basically, we can stain for nearly all of these things. So that's why people are always trying to look for surface markers which somehow can identify cell, because that's kind of the easiest way. Intracellular cytokine is good, sorry, intracellular staining is good, but it is very cumbersome and it requires many more number of cells, and it is also longer, but it can be done. It is not that it cannot be done. Especially for cytokine, it has now become more or less more routine in many ways. This is basically is what it's showing you, that you can look at the cells by intracellular staining. So, I told you that cytokines will accumulate inside the cell and secretion will stop. So that's why you can look at by flow cytometry. This is an example of here. So you have CD4 T cells you have sorted from PBMC, and this is PMA and ionomycin are non-specific stimulate. These are known as mitogenic stimulation. Basically, these cells will stimulate T cells without the TCR. It doesn't require that you need to activate the cells with TCR. They just cause non-specific stimulation. And Golgi stop is basically something which you add to prevent secretion of the cytokines. And you need to permeabilize because you need to have, you need to go inside the cell because you want to look at intracellular staining. So there's a buffer known as fix and perm. You can make it also, but basically it has a mild detergent known as saponin, and that is what. And then you use an antibody for interferon gamma, IL-17 and IL-4 because you want to see if it is a Th1 kind of an immune cell or a T cell or a Th17 or a Th2. So here, this is interferon gamma and IL-17. Here, you can see that there is some IL-17 being secreted, and most of the cells are interferon gamma positive. Hardly, very few cells are also double positive. When you look at IL-4, again, you see again, quite a number of cells are interferon gamma positive. Some cells are IL-4 positive, but you are also seeing double positive cells. So this is simply showing you that you can use it to detect cytokines. And here is an example of one of the patients where it's the patient is producing high levels of, here the number of cells look less, but the percentage of cells which are producing interferon gamma is about 26%, 25.8, while in the control it's only 7.6. So more of interferon gamma is being produced by the patient cell. same thing is shown again here on activation, that it is producing high levels of interferon gamma. So this is an example of the lupus where you are looking at activated T cells, because 25 is an activation marker. So in the patient, there's less activation. In control, it looks as if there's more activation. But when you look at this, so CD25 is, how do I say, CD25 is also a marker for activation, but very high expressing CD25 cells. So CD25 is expressed in this way. It's intermediate and some cells are very high. So the T regulatory cells express very high levels of CD25. Very, like on activation, it is in the intermediate stage, but there is a very high, which is there in T regulatory cells. So basically here what they are trying to show based on the CD25 is that you have natural Treg. Basically, don't worry about natural Tregs and induced Tregs, it will confuse you more. The number of T regulatory cells are lower in the lupus patient, and that's why there is more immune activation. That's what they are trying to show here. And then these are all different deficiencies which have been identified. Some of them I have already shown you, and they are looking at regulatory T cell numbers in these diseases. So it's kind of down in the X-linked syndrome, and when there is deficiency of CD25, again it is slightly down. In all these cases, it is down. In this Viscott-Aldrich syndrome, the numbers are normal. But in this case, the function seems to be more of a problem. The numbers are normal, but the function is not at par. And these are also all different kind of deficiencies. So, yesterday I had mentioned that if the cells are making more IL-23, they will that you will get more of a Th17 development. So if you have this deficiency, there is impaired development of Th17, and this has been shown in patients. So this is another thing is here. IL-17, I told you, that is important for fungal infection control. If there is a deficiency here, you see a lot of candidiasis, which is a fungal infection. So that's how people have come to know that how, many of these things have come from patients or mice, that where these cytokines are useful and what are their functions. So anytime you see an abnormal function, it basically can tell you a lot of different things. Okay. So this is an example where you have this JAK-STAT pathway. Different molecules in the JAK-STAT pathway are either somehow or the other absent, and then you see these manifestations. If this is absent, you increase susceptibility to infections. And then the defect is production of IL-12. So similarly, like all these different molecules are leading to some kind of a defect in cytokine, which you can use flow to measure. So this is basically, it tells, this I have taken from NIH's website. It tells you all the different kind of deficiencies which are occurring. So wherever you have a number, so basically, there can be deficiency in the bone marrow, that the cells are made, they cannot exit. There can be deficiency that they are unable to migrate. There can be a deficiency they are unable to, differentiate. So basically, wherever there is a function involved, there can be a deficiency, right? So this just shows, kind of that. And this is basically your flow cytometry. So now, in fact, newer machines have come where people are using antibodies which are labeled with the metals, and the machine is known as CyTOF, where you can go up to 40 to 50 markers, and instead of flow-based detection, it is a mass spec based, because it's a metal there. So they vaporize and then they can measure different kind of these subsets of cells. So that is a more recent addition which I haven't used, so I didn't want to talk about it so much because, but I'm seeing that it is becoming more and more useful. I've just started seeing a lot of papers and there people are identifying newer subsets. I see it to be like I see a lot of people working on cancer. They seem to be using this cancer, this cytoph, because from one sample, if you can stain with these say 40 antibodies, whatever, you can find out if you take a tumor suspension, you can find out tumor cells, you can find out immune cells in that, you can find out what kind of immune cells are there, the matrix cells. So with one sample, you can get all this information. And then based on that, you can draw conclusions that, okay, maybe I should do this or I should use, I see too many numbers of T regulatory cells in this patient. So that means there is a major inhibition. What should I use? So I see that it is slowly becoming useful. Earlier people were more focused on genomics to get this kind of an information. With the cytoph, the advantage is that you're looking at the protein level. With the gene level, sometimes it is that maybe there's no translation. So that's why the cytoph, and now I've heard that there is a new machine. This is a cytoph, I think, is made by Fluidim. They have come out with a new machine where they are trying to take sections, the original sections, and they pick up cells, and you can even look at those cells and see. So the advantage of doing this is that you know where the cell is coming from, which part, So when you make a suspension, you no longer have the anatomy or the structure there. So you don't know if it is from the tumor microenvironment stroma or is it from the tumor. But when you do things like this, you know where the cell is coming from. So usually a combination of like flow cytometry and microscopy sometimes are also useful. In fact, there is a flow cytometer which is from, what is it known as? Image stream. What it does is, it does both. It takes pictures and it does flow cytometry also. So you can get the localization of the molecule. Is it, how much of it is extracellular? You can see the picture basically, what you would do in the microscope, and side by side you can get the plot also. So every day, this whole field seems to change. We have a special conference every year in our university, which is on flow cytometry. And so many vendors come. Each year I see some new kind of a flow cytometer, which has some unique function, like either the compensation, they have done something so the compensation is fixed. They've changed, like a lot of engineering is going into this area. So we'll see, I think, many more advances. But the machines have become much more expensive also. Yeah.
Count of normal and abnormal cells?
You can. If you know the marker, you can get the number of abnormal.
So can we use this flow cytometry in case of liver cirrhosis? In that one, we are just, I just want to get the normal cell count and abnormal cells count, means damaged cell count.
So do you know what kind of a damage is there in that they are dying, these cells, or how do you say that they are abnormal?
They are not dying, but they are not functioning as they are supposed to.
As they are supposed
to. They are just malfunctioning about that.
So you know that there is a functional defect.
Yeah.
Now you want to see with flow cytometry if you can. So then you need to look at maybe certain markers for those cells. If these are macrophages, like if they are Kupffer cells or whichever cell type you are focusing on, you need to look at the markers which are there and see if there's any expression difference, which can tell you that in case of liver cirrhosis, this marker is less expressed in these patients. So that will be one way of and then you look at, you already know that they are malfunctioning. So then you associate that, the difference in expression with the malfunctioning. Then you can make it a test in a way that, okay, when you have this, suppose it is fibroblast, and I say that you're not getting this FBAP or some kind of a molecule which is very common in fibroblast, you find less of an expression of that. So you can, and these cells then are non-functional. But you see this in all liver cirrhosis patients. Then you can make it a test. Okay, when this is down, you can predict that these cells will not be functional.
Basically, it will gives only the count about that cell or size and shape about that cell.
It can give you the size, shape no. Shape will come in the microscope. Size it can give you, because the FSC, SSC, the forward that is based on size. So if these cells are bigger, they will show up as different. Like you can see the size that they are different, or smaller also you can see the size. So size you can see, you can look at different phenotype, or if the function, if there's a specific function, if it's a cytokine secretion, or you think there is some transcription factor or some kind of a signaling which is defective, you can also look at it by flow. That can also be done by flow cytometry. Like if it's not producing something, then it has to involve some kind of a signaling for that protein not to be produced. Then you can look at that signaling molecule as a diagnostic.
Thank you.
Okay. So this is basically flow cytometry. There are other assays also, but we'll take a 5-minute break and then we will think about it if we need to talk about that or not. ओपन करना भूल गए.